It's probably very, very difficult. If I could take bets, I would take anything from 2020 to 2800, highly likely. But the things that we usually go for when we look at a patient, at an OCT of a patient with macular edema, would be whether they have the photoreceptors, the ellipsoid layer intact, the cyst, hyperreflective foci, and also where the inner layers of the retina are distinguishable. We need to know the anatomy of the eye. Supposedly, we already know that, the anatomy of the retina very well. And we need to know what is exactly the correlation that we have with the normal OCT. Because how many of you do OCTs on normal people? Most of the time, there's something missing. So we need to remember this image so we know exactly where we're standing. Whatever is missing may be the reason why the vision is not picking up. But diabetic macular edema has a major problem. The correlation between structure and function is very, very poor. What I mean by this is you may have patients with a lot of edema and very good vision, some others with little edema, little thickness, and very poor vision. And let me show you a couple of examples of these. This patient here has 200 microns in thickness and about 20-25 of vision. This one here also has 200 microns, but vision is a lot poorer. On this spectrum, we have patients with good vision and 200 microns, but you could also have good vision and about 600 microns. So you can see this is not a linear relationship where all the dots are sitting very close to the midline or within the confidence interval, but actually is a little bit more scattered. That means it's very difficult to establish, just by looking at the thickness, what the vision of the patient would be. But it gets actually worse. When we do the treatment, what is it that we expect? What is the magic that we expect to happen, whatever treatment you do for the patients? We want the retina to go thin again and vision to go up. And that would be this case here, where patients have a thinner retina and better visual acuity. But there will be a lot of patients which will behave in a paradoxical way. That means thinner retina and worse vision, and some others with thicker retina and better vision. So most of the time, if we try to guesstimate what will be the response just by looking at the OCT, we will be fooled into believe that something has happened when actually maybe the opposite happened. We go back to our patient saying, look at your OCT, it's great. It has decreased 150 microns. And the patient looks at you saying, you must be nuts because my vision is actually worse. So what would be the explanation for that? There's a paper that has been published a couple of days ago, and it's some work done by Ursula Schmidt-Erfurt looking at several patients with DME, and they support this idea of this very, very poor correlation, even with newer techniques of OCT. So the correlation between just thickness and visual acuity in this type of patients is very, very poor. So the one thing that we may look at, as I said before, is whether the patient has an ellipsoid layer. That will be vital because they need photoreceptors to have vision. But also we may look at something that is called DRIL, disorganization of the retinal internal layers. It's getting more and more popular that people know what the term means and they are able to identify it in the patient. And that is going to help me establish whether the patients are improving or not the vision, or they will improve their vision or not. Let me show you a simple example so you can have the paper there where this information can be further read. These patients here, both of them have macular edema. This macula is actually thicker than this one. Photoreceptors appear to be quite good in both patients. But this one, if we focus on the central 1 millimeter, we will see that this area is totally grayish where no layers are distinguishable, while on this side, all the layers are very, very clear. That means half of the macula is fine and the other half is not that well. So this patient has a 50% DRIL, and we may say to this patient, even if we get rid of the fluid, vision may not pick totally up because this means damage to the inner retinal layers. While this patient here has a massive cyst, and you can see the layers very, very clearly. So when you treat this patient, even though he's got larger cysts under the fovea, his vision will pick up faster, although at the beginning, the thickness is actually worse than this patient. Let me show you another example. Patient with massive cysts, photoreceptors are good. You do the treatment, whichever you like to get rid of the fluid. You get rid of the fluid, but you can see that the center part of the fovea, it's all gray, no layers are distinguishable. That means there's a lot of DRIL. This patient has reduced the thickness, but probably gained no extra vision. While this one, with massive cysts, exudates, intact photoreceptors, and the inner layers of the retina, very well distinguishable, has no drill, and as soon as we treat the patient, it's going to get better and vision will pick up. So it looks like when we do an odds ratio for chances of improving vision, drill appears to be, for several patients, all eyes, patients with current edema, patients with resolved edema, the best predictor for gaining vision after treatment. So that is actually better than total thickness and presence of cysts in the retina. So it looks like maybe this poor correlation between thickness and vision could be explained by we should look at the quality of the OCT rather than the quantity of the total thickness. So if we look at the quality of the OCT, whether there are photoreceptors or not, whether there's drill or there's no drill, we may better guesstimate what will be the outcome of that patient rather than just looking at thickness, which seems to be a very, very poor predictor for good vision. So once that we have identified the OCT, look at the patient, explain things to the patient, we need to make our decision, what is the treatment that we are going to have with this patient. Protocol I is one of the DRCRNet trials that explains exactly what is the outcome when you use the more typical treatments that we may have, which is having focal laser to the macula, having the use of anti-VEGF with early or late laser, or having steroids, in this case is triamcinolone, but I will show you something regarding the dexamethasone in the future. So we may look at these results in different ways. One simple way would be, this is the best treatment, this is second best, third and fourth, and that's it. I just go for the first, the best one that gains 10 letters. But what if I tell you something in the opposite direction, like this one? I tell you that this patient treated with laser at a very reasonable cost, just 3 sessions of macular laser, managed to gain 3 letters. While these patients here, they received 14 injections over 2 years at a much higher cost and actually having to come very, very often to the clinic. And the only difference between average vision between these two groups of patients is just 5 letters. And you would say, okay, that sounds reasonable as well. The problem is, these trials should not be looked at just by the efficacy, but also the amount of efficacy and amount of harm. You know, there is something in statistics that we can do that is called number needed to treat, but we can also calculate the number needed to harm. I usually put a simple example. What if I come with a pill that I tell you it cures diabetes completely, okay? And you take the pill, whatever diabetic patient you may have, takes the pill, 80% of them get totally cured of diabetes. You should ask me what happens to the other 20%. And my answer would be, they die. Would somebody like that treatment? Is it a treatment that we will be able to sell all across the world? Probably not. What if I come with another treatment? 20% efficacy to get rid of diabetes for good, 80%, nothing happens. They carry on as their life as they were the day before. So that is going to be a much more successful treatment, although they have 4 times less efficacy. It's only 20% versus 80%. So that's the extreme example that we shouldn't look at just efficacy, but we should look at both sides of the coin in this case. So we can look at this trial, which is not exactly a fair comparison because we don't normally do either early or deferred laser. That's something of the past probably. Triamcinolone is not the steroid of choice these days, and laser we can do it better than that probably. But if we look at these images from a different perspective, we can compare the great success, that is gaining 15 letters or more, versus great damage, which is losing 15 letters or more. This is something that will affect the quality of patients, quality of life of these patients dramatically. So how many patients will gain 15 letters or more when treated with anti-VEGF? It will be about 25–30%. Do these other treatments work? They do work really well. Look, 15–20% of patients may have a benefit on gaining 15 letters or more when treated with laser or steroids. But when you look at the other side of the coin, how many patients lose vision because of the treatment, is higher the proportion of patients losing vision because of what may have happened. Scars to the fovea, enlargement of the damage to the retina due to the laser, something related to the long-term efficacy of the steroids, and very, very rarely you will have treatment failure with patients treated with anti-VEGF. So the reason why mostly everywhere worldwide the use of anti-VEGF is considered the first line of treatment Of course, there are subpopulations which you may select other treatments as a first line of treatment. But for the great majority of patients, usually anti-VEGF would be the first drug of treatment. It is because there's a very good correlation and very good proportion between the amount of benefit and the amount of damage. And that is graphed here, represented here in this table very clearly. So when you use anti-VEGF, great benefit and very little damage, while the other ones will have a proportion of patients losing vision compared to the amount of patients gaining vision. So this is the reason why everywhere people would prefer to use anti-VEGF as a first line of treatment without looking very much into the case, although in some cases you may use something different. The only head-to-head trials that we may have today, because the data that I was just presented was done with triamcinolone, and these days very few of us use triamcinolone, although some carry on use it because of the cheap cost of it, is this study that is called the Majore trial. And it compares patients treated with dexamethasone implant every 4 or 5 months versus ranibizumab used on one loading and the rest PRN. Although this is an unfair comparison, this is a trial that is doing an unfair comparison. It was an Allergan design trial. It is unfair because you're measuring the endpoint only 2 months after the last steroid injection, and you should actually measure it 4 months later, not 2. And usually we know that the use of a loading of 3 loading is actually better than doing just 1 loading. So even with this unfairness of the trial design, and trial design is everything, you know, if you do a proper trial design, you can show whatever. You can show the opposite, or you can change things. Even with this unfair trial design, for the same baseline visual acuity, the visual gains were much better for those patients treated with ranibizumab. Although you need to do more injections, you need to see them more often, and you know, it's a different treatment, but the visual gains are much, much better in these type of patients. But even if they come in the future with a new steroid, let's say they come with a steroid that induces no cataract, visual gains are as good as any visual gains that you may get with anti-VEGF, I would still prefer to use anti-VEGF for the treatment rather than steroid, for a very good reason, which is the following. When you treat patients with DME, you aim at targeting the VEGF, which is the molecule responsible for the increased leakage of this blood vessel, okay? So when you're targeting the VEGF for DME, you're also targeting it peripherally for the development of PDR. So it's one of the molecules that affects PDR, but it dramatically changes the way the PDR responses. What I'm saying is, when you have a patient with DME, there are high chances that the patient may have some form of diabetic retinopathy. When you are treating your DME with anti-VEGF, you're having both an effect on macula, looking at the macular edema, but also an effect on the retina, having some effect on the diabetic retinopathy of the patient. And this is a very simple example of that. This is a patient that showed up in the clinic with high-risk PDR. He had macular edema. His only complaint was poor vision. We showed him the photos, explained the whole thing. He was entered into a trial using anti-VEGF for treating macular edema, and after 36 injections, this was the Right and Right study, after 36 injections, he ended up with no macular edema, good vision, and he got rid of some trouble to the back of the eye, right? So based on this was sort of a pearl within the trial. We found patients having regression of diabetic retinopathy while they were being treated with intensive treatment for macular edema. Intensive treatment means a lot more injections than what you and I may do on a clinical patient. So we went and explored further and looking at the type of patients where we want some regression to happen. Do we want regression on these patients? They don't need regression. They're doing well. Do we need regression on these patients? Well, this one will receive PRP soon, and these guys are just waiting at the entrance of the operating theater. So which are the ones at which, when they show macular edema, we would appreciate it if we can have some sort of regression of diabetic retinopathy? And that case would be the 4753. Am I doing something wrong? No. It's the 4753, the advanced NPDR cases. If these patients had macular edema, my treatment of choice would be almost mandatory anti-VEGF. I could treat these patients with anti-VEGF or steroid. I would look at other factors, pseudophakic or not, responded to pressure or not, pressure responded to steroid or not. But these guys, if I treat the macular edema with a few injections, these will benefit dramatically from a regression of diabetic retinopathy. So we did a randomized further analysis of these patients, and we found that at this level 4753 is when some dramatic changes to quality of life and driving difficulty start going down. Driving difficulty could also be related to near-distance working ability. So these patients are at working age, and they may benefit from treatment at this time point. We went back and looked at all the patients that were being treated with level 47 and 53, and this is level 47 and 53 at baseline, and this is what we found out, that when you treat patients with this level of diabetic retinopathy and macular edema, as soon as you do 3 injections, you're going to have 1 every 3 patients showing this regression of 2 steps. And at 6 months, you're going to have 50% of your patients having a regression of 2 steps. Then, if you want to carry on, you're going to need to do continuous injections, as this trial did. But 3 or 6 injections is something that we may normally do on a diabetic patient, and that means 1 every 3 and 1 every 2 will feel this regression in the retina, which will be a good benefit for them. Listen carefully, I'm not saying that the treatment for diabetic retinopathy is the use of anti-VEGF. The treatment for DR, progressing into PDR, would be PRP now and for a good while. In the US, doctors are allowed to use this treatment for diabetic retinopathy even with or without DME, but we are far away from the US, both you and I. So the situations are very difficult and different. But even if we manage a patient really well, we appreciated the DR status, we consider that using anti-VEGF as a first line of treatment would be the best option. We start treating several patients, and after 6 months, we call them all back into the clinic. We do a good quality, spectral domain OCT, and we see that 40% of them, they still have fluid in the back of the eye. And we have all been through this situation, having almost 50% of your patients saying, how come I've been doing such an effort? The patient has been doing such an effort, 50% of them are happy with the treatment, and the other 50% are not that happy with the treatment because they may have a thicker retina than expected. So we have several options here. One option would be carry on injecting. Second option would be switch for another anti-VEGF. And the third option would be add steroids. So let's go and quickly review the evidence for this. In this paper, the Bresslers, they do an analysis of the persistent macular edema in protocol I. And basically what they suggest is that the way forward should be carry on with your injections. Whatever you're doing, just carry on. Fluid would eventually go away. And actually, don't be too bothered about fluid at this stage, because they have seen that throughout 3 years of treatment, patients with and without macular edema ended up having more or less the same vision. So it looks like after 3-6 injections, what ends up mattering most is vision for the patient rather than what is the total thickness of the OCT. So they say, don't worry too much about thickness, fluid will eventually go away, keep injecting, fluid will go away. Okay, so that's your first chance, your first option of treatment. If you want to carry on, this is the evidence that supports your decision. What if I want to switch? Have you all read several papers about switching? They all agree in the same thing. If you switch from A to B, B to A, whatever drug you want to call A or B, you will have the effect of dropping 50 microns in thickness of the OCT and gaining 3 to 5 letters. So that seems like a good thing, like a positive benefit for my patient. But all these trials, they make one common mistake. They switch every single patient. They don't leave a subgroup as a control group to see what happens. Let me show it to you with a very, very simple graph. You have a group of patients with macular edema. You start treating them with drug A. There's a subgroup of patients that do not respond according to whatever you think they should be responding. You take them out and you start treating them with drug B. You will find papers starting with A, B, B from A, whatever combination you may find. And then, after doing A to B, you see a positive effect, increase in vision and drop in thickness. And you will be highly likely fooled into believe that that was due to the change from A to B. But you have done 2 things here. And in science, when you do 2 things and you have one result, it's very difficult to attribute that result to either one or the other thing that you have done. So here you can see that we most likely would be thinking that B was the big change, but actually patient received a number of injections up to here and a higher number of injections up to here. So if there are any late responders, they will pick up vision and keep dropping the thickness of the OCT. And it will not be because of the change, but it will be because they receive further injections. The ideal switching study, which is not available out there, is the following. A study in which you grab patients treated with A, you take the underperformers, you take them out, and you randomize them to either receive treatment B or A. Then you will evaluate the results that you get from this, compare them, and if there's a true difference between them, then it means that switching from A to B worked, or maybe it works just staying in A. And if you do that, you will have to do the trial again, starting with B and then randomizing again, because you want to see whether switching works both ways, okay? This trial, unfortunately, it doesn't exist in ophthalmology with the use of anti-VEGF. It doesn't exist. So there's a paper done by the group of the CAT study, and what they say in this paper is that a control group maintained on the original agent should be included in studies of switching to make a valid conclusion. So you need to make sure what is switching and what is continuous injections. And no trial has done that. So what they've done to prove their point is the following. They created these hypothetical switching criteria, some basic stuff. According to these criteria, they flew back in time. They grabbed the data from two trials, the CAT trial in wet AMD and protocol I in DME, and they saw the patients, what was the performance at 3 and 6 months. And they looked whether they would apply that criteria and switch them if they were there at the time. Something that they didn't because none of these trials did any switching. So we're only exploring the, you qualify for switching, but you don't switch. And this is what they end up having. Patients that would qualify for switching, at the top you have neovascular AMD for 3 and 6 months, and this is DME for 3 and 6 months, and in this case this is vision. Gains of 3 to 5 letters in vision, and drops of 30 to 50 microns in the OCT. So it looks like these trials show that patients qualified for switching, they were not switched, and over time they ended up gaining vision and dropping OCT. Most likely what we see is just late responders. So there's no strong evidence for option number 2. The first one is keep injecting, and there is some post-hoc analysis that supports that. The second one is switching. Doesn't seem to be any evidence supporting the switching of patients. And the third one that we decided to do would be addition or switching to steroids. For that, we can look at the evidence from the Protocol U. Protocol U is another DRCR-NET trial, and basically what they've done is compare the combination therapy that is ranibizumab plus the dexamethasone implant versus just ranibizumab. They did it in a way, if you have a patient that you've done 3 injections already and still have fluid in the back of the eye, you may invite the patient for the enrollment part of the study. That means receiving 3 more injections. Up to here, they will be receiving up to 6 injections. This day, you evaluate them, and if they have fluid in the back of the eye, you would randomize them to either carry on ranibizumab all the way, and receive 2 dexamethasone implant every 3 months, or just carry on with the ranibizumab and simulation of the dexamethasone implant. So either combination or not, but heavily treated with ranibizumab as a base, and on top of that, the same day here, you do the dex implant, and here you do a dex implant as well every 3 months. Not 4, not 5, not 6, okay? And these are the outcomes that this trial gave. These are the visions that you gain if you carry on with ranibizumab after determining that your patient at 6 months has a lot of fluid, and this is the vision that you get when you treat your patients with combination of ranibizumab plus the steroid. So it seems to be that there are no really clinical statistically significant differences by doing this. The only difference that we found in this trial is that the drop in thickness for ranibizumab is 60 micron and is 110 for those patients receiving steroids. So it means you would do a big effort on the pocket of the patient, on the eye of the patient, because we'll be receiving a lot of injections, and all you're going to get is 50 microns less on the OCT, because vision is going to be the same. So it's up to you to decide whether that is a valid, if you think that a drier retina is going to be better for the future or not. So the 3 options that we have when we manage patients with DME that have still fluid after 6 months of treatment would be keep injecting, there's evidence for that, switching, no evidence for that, and adding steroids. There's evidence showing that no visual benefit, but maybe some benefit regarding the thickness of the OCT, which we still don't know exactly what is the meaning of that, or if that is a positive thing or not. So what I wanted to show you here is that we could possibly split all our patients with diabetic macular edema into 2 groups. The happy group, the ones that will respond quickly, drop the thickness, increase vision, they love us, they come back with presents. And the other group, the one that will come with that face saying, do I need another injection because I'm not getting any better? And those are the ones that you will be struggling with. You will have the voices behind your head saying, you're doing wrong. Something you're doing wrong. You need to switch. You need to add steroids. You need to switch to steroids because as if, the ranibizumab was reducing the effect of, you will find lots of explanations in the jungle of papers out there and the jungle of experts that we live in. But the truth is that revisiting all this data, these are more or less the options that you're going to be facing with. And this just comes to show that you're not alone. Even the best experts worldwide face trouble when managing the small subgroup of patients with diabetic macular edema that do not respond very well to any of the treatments that we may have. Some may ask, what if I had laser to these patients? And the RESTORE study compared ranibizumab versus ranibizumab with laser and actually added nothing. All you add is suffering, wasting time, and maybe some scars to the back of the eye. So you're not alone. Some patients with DME are very, very difficult to treat. Thank you very much. Shall I move to the, or we do questions now? We do questions, right? Okay, excellent. So I'll do my second talk. It's all being done for me. Okay, so the purpose of this talk is to discuss something that it started happening in your country and in my country as well. We live very similar realities. We see very rich patients and very poor patients, and we have to make sometimes dramatic decisions on the way we treat them. And we have governments that want to make sure we lower the cost as much as possible. And this is a wave that will come in the future, okay? So I'll clarify this for you during the talk. Age-related macular degeneration has a very good name because it's an age-related condition. And we can see in all these population studies that these curves of incidence of AMD, they go straight up as soon as we get near the 90-year-old mark, okay? That means as soon as we start living beyond this line, most patients will have some form of AMD, be it a couple of drusen or being a full-blown geographic atrophy in both eyes with membranes on top of that, okay? So this is gonna get more and more common in our population. And the life expectancy, let me tell you, is growing, and it's growing really fast. We were discussing before with some colleagues saying, in the past, people used to live such a quiet life, retired from pollution, not directly linked to technology, and my answer was, life expectancy was 32 years. So clearly, stress is not doing any harm to us. Maybe we're getting a bit of stress, but at the same time, we're getting great medical treatments, and we're prolonging life very, very quickly. Probably the biggest change from here to here for the 1950s was the availability of drinking water. That was the major strike for medicine, separating the sewage from the drinking water. And that increased life expectancy in the 1950s up to 48 years. This is the worldwide average, okay? And these days, it's above 70 worldwide average, several countries going beyond the 80-year-old mark. Soon we're gonna have, and these things are growing fast, and I know that this country is growing almost at a 10% rate economy-wise, and that will have an impact in the life expectancy. So be sure that your clinics are gonna have more and more patients, and always remember that this is a bus that people get in, but nobody gets off. Or do you have ever discharged a patient with AMD saying, well, I've done 17 injections, now you may go home comfortably. If there's any trouble, you give me a call. That doesn't happen, right? So we inject them until they're not longer with us. We never say that to them. When they ask me, is this treatment going to be forever? I say, no, it's not going to be forever. It will be until I have something better to treat you. That's the word I use. What about diabetes? There's a lot of diabetes. We have tried just some great dessert out there, and that means we're going to have even more diabetes. So diabetes is everywhere, but there will be even more diabetes, okay? So we will have a lot of these patients. We will have better treatments as well, and more available medications for these patients. Have we been making an impact on wet AMD? This is the natural progression of the condition, And this is what we have achieved with the use of anti-VEGF. Finally, gaining visions and maintaining visions, albeit the long run for patients with age-related macular degeneration will be ended up having some form of atrophy that will affect vision. And what about diabetic patients? This is exactly the same case I showed before. We have never seen diabetes go away from the eye. Never seen before. It's only with the introduction of anti-VEGF. I'm okay, yes, you have to do a lot of injections. Not on every single patient this will happen. But this is something striking. This never happened before. Younger doctors that have been seeing patients for a long period of time can tell us, have they ever seen patients having dramatic regression of diabetic retinopathy like this? Never before, probably, okay? So this is, we're making a high impact. We need to make sure we treat more patients, and we treat them more often, and we have a lot of trouble still. My situation is very similar to your situation. And this is a description of that. This picture could have been taken anywhere in India, or anywhere in South America, believe me, okay? So we live in a situation in which some patients have access to everything, and some others have access to sometimes not even water, okay? And it is important. Everybody talks about healthcare, poverty and wealth, the economy, the impact that this may have on people's lives, and so on. And we all want good treatment at a low cost. We all want that. And let's review what would be the ideal treatment for our patients. Ideal treatment would be highly effective. And these drugs are highly effective. If you calculate the number needed to treat for maintaining vision for patients with wet AMD, it's actually 1. Number needed to treat is very near to 1. You want to calculate what is the number needed to treat for patients taking Lipitor, for example, statins, it's 99. 99 patients need to be treated to save 1 of them. Okay, so all these other drugs that are out there treating a lot of patients, they need to treat a lot of patients just to save 1. Here, for wet AMD, maintaining vision, 95% of the patients will maintain vision on treatment. May not gain, 30% of them will gain only. So we have a highly effective treatment. Safe. If we do it properly, injections are very, very safe. You just take all the measures and make sure everything is clean. The ratio these days in Europe, the reported ratio of endophthalmitis is less than 1 in 10,000, okay? And probably somebody messed it up that day, highly likely. Cost efficacy. Cost efficacy is extremely important. And to calculate the cost efficacy, most governments in fully developed countries, what they do have is organizations that they would look at cost efficacy, totally independent of any other bodies. They're not part of the Retina Society, they're not part of the pharmaceutical industry. Independently, they would look at cost efficacy. For example, NICE, the National Institute for Clinical Excellence, that we have that there is in the UK. And to calculate that, you need 2 numbers. 1, units of money that you need to buy the drug, and the second one is what is the efficacy. Is it 100% of another drug? Is it 110, or is it 85? You need to know exactly what the efficacy is to do a cost efficacy calculation. We need drugs that are accessible. Some drugs, we have some diseases in Argentina that are very easily cured with a very cheap pill. The pill is nowhere to be seen. Nobody wants to manufacture it because it has no profit, okay? So treatment needs to be accessible. Easy to administer, and although at the beginning doing an intravitreal injection was a dramatic event, these days we do hundreds in a day, and we just go off for lunch not saying, oh my God, what have I done? And at the beginning we were doing 1 injection and not sleep for 3 days. Now we do 100 and we forget about them 5 minutes later, and the patients as well. Tolerable for the patient, they cope with the treatment really, really well. So this is what an ideal treatment should be. Let's review what is a biologic product. There's a long list of examples of biologic products, but basically we could say that a biologic product is an agent that is used for diagnose, prevent, treat, or cure diseases, and basically it just comes from a living tissue. It's not produced by mixing a couple of fluids, but it's actually produced by a living tissue, be it a line of cells, an animal, whatever, okay? So that is what a biologic is. On the other hand, we have a lot of macromolecules, and macromolecules are these. Aspirin is a macromolecule. Very light, very simple structure, still very effective, and contrary to the macromolecules, or very complex molecules, this is adalimumab, which is a very extensive, fully human antibody used for several indications, one of them, the posterior non-infectious uveitis. It's extremely expensive, and I heard that it's not available here in India. But it's a heavy, heavy molecule, and it's very complex. It means if we change a few amino acids here and here, maybe the performance of the drug will change dramatically. Generics are copies of micromolecules. Easy to copy, simple molecules, easy to prove that they are exactly the same. You just drop 2 pills in a glass of water, they dissolve more or less in the same time. That's as far as you need to go regarding testing to release a generic, and the purpose of generics will be to reduce the price from the original drug, okay? Some people prefer the brand, and they will pay slightly more for the brand. Some people are happy with just taking the generics. They are happy that the government have approved them, tested them, and made sure they work exactly in the same way, saving a few units of money, whatever country you live in. But now we come to biosimilar, and both countries, India and Argentina, are going through an introduction of biosimilars into the broad offer that we have of treatment, official treatments and unofficial treatments, and now we have something in between, which is the biosimilar being introduced to the market. We are living exactly a very similar situation. What is a biosimilar? We said that the biologic is a biological product, so a biosimilar is a biological product that is highly similar to and has no clinically meaningful differences from a reference product. That means it's a copy, a generic of a biologic. We call it biosimilar. It needs to be highly similar and have the same efficacy, okay? So it's more to do not with the looks, but with the performance of the drug. Minor differences may be accepted if they have no clinical impact for the patients. And this is not a definition that I have just invented. This is the definition from the FDA. And we could say that the FDA is the most stringent regulatory agency to evaluate drugs that come out to the market. That is why, for example, all trials that come from the FDA would be the twin trials. And they do twin trials because they want to make sure there's no variability, and they can account for viability by doing 2 trials. Example, Ride-Rise, View 1 and View 2, Hawk and Harrier, all these twin names, it means it's an FDA trial, okay? And FDA was the agency that managed to have no cases of thalidomide deformities in the US, because they did not approve it. They say, we're not going to approve a drug to be used on pregnant ladies unless we have some sort of testing, at least on animals. So all the cases that we have seen are worldwide, mostly not originated from the US. Some were imported into the US, you know, people taking medication abroad and then coming and giving birth in the US. But it's because the FDA was so stringent that that didn't happen. So we can take for granted that the FDA is the highest standard for approving drugs. A biosimilar may achieve a very high status, which is the interchangeable status. When a biosimilar reaches that point, the government will replace the original drug automatically for the interchangeable biologic. Unfortunately, not a single biosimilar managed to achieve this status, which means the government will replace it automatically. Why? Because you need to do a very large and complex clinical trial in which you split your population into receive the reference product, the original biologic, and the other group will be switching back and forth from the original to the biosimilar several times, just to make sure the efficacy is the same and there are no safety signals. If any biosimilar achieves this, it will be considered better than because it will have a lower price and it will be totally replaceable. Nobody in any medical specialty achieved this for the FDA yet, okay? The importance of this is that sometimes biosimilars may not be well designed, and they may induce something that is called immunogenicity in the patient. It is interesting that the micromolecule market is the largest part of the pharmaceutical industry, but you can see that the biologics is a big chunk of it growing, and these are highly very costly medications to treat patients, but they have a dramatic impact in the quality of life. And that is why the biosimilar companies are trying to copy these things. So let me put some simple examples regarding the micromolecules and the biosimilars. Let's say you go to Retina in Vienna in a few weeks, and you decide to buy a pair of football shoes for a member of your family. You go and check the original one, and they cost something like 400 euros, very expensive. You say, I'm not going to pay that money, particularly if it's for a boy, they're going to grow out of them very quickly, and you buy the copy version of it. Somebody with a blanket in the corner of that shop, they will sell it to you for 20 euros. What would be the problem is this simple product is not the same. I mean, is the hologram inside that kids look for, or what the performance would be? Would it be too much of a trouble if this doesn't work? Clearly not too much. Nobody's going to die by using fake football boots, okay? So a micromolecule, if it's slightly better or slightly worse, it shouldn't be too much of an issue, okay? And usually it's not. But what about if we're talking about more complex molecules? And I'm showing a very complex molecule here. This is a complex molecule that has a very good performance, and this is a molecule, complex as well, not as complex, looks similar, but what would you do to make sure this performs exactly in the same way as this? What would you do? You just take it to the track, right? And you do a few laps. You see the speed, maximum speed, ability to turn the bend very quickly, and just make sure you get off the car and you had a lot of fun and you didn't die. And this one, you know, sometimes try to take the bend at the same speed as this one, and you may just turn it over and have some damage to your safety, okay? So the way macromolecules, big complex molecules, should be tested is by doing a comparison between them, okay? Also, we need to look at where they're manufactured, and highly likely the Ferrari comes from a lab like this, that more than a factory looks like a lab, actually, how they do it, and the other car probably comes from a place like this, okay? And just to quote an example, and this is not a talk aimed at saying anything good or bad regarding any biosimilar in particular, but I just want to quote, there were sprouts of inflammation here due to the use of a biosimilar that is available here, and these several cases of inflammation repeated again 2 years later. So something happened, they did reformulation, and then what happened with the reformulation? Did it go back to the original formulation and then it was released again? Nobody knows, but the second wave of inflammation, we should call it, happened again. So an inspection from the FDA was done, and they found several things. Some of them may be minor, but some of them could be quite interesting, like a building that is not free of infestation by birds. That doesn't look like where I want my drugs to come for my patients. Okay? And we said immunogenicity is very important, and if the drug is not pure enough, it may be efficacious, but it may generate an immune response in the patient using the drug. And that means maybe after developing antibodies against that drug, when we use the original drug again, it doesn't work, because the body has developed antibodies against that drug. And I'm saying maybe, because this is very difficult to test and prove. But biosimilars are not a bad thing. They are a great thing. In the future, patents will fall in the US and Europe, and a lot of biosimilars will come. Let me tell you, everybody's copying everybody. Sandoz, which is a part of Novartis, is copying the Humira from AbbVie. Genentech is copying Pfizer. Pfizer is copying Amgen and Merck. Everybody, even Samsung, which wasn't a player producing drug, now they are producing biosimilars. So in the near future, I'm talking about 3 to 5 years, we're going to see a lot of these drugs floating to the market. So I'm not talking particularly about the one that you have these days now, but in the future, we need to make sure that governments approve these biosimilars following the right pathway and not just a quick way out just because we need a drug with a lower price, okay? And there's some definition that we should look at, and one of them is the following. Maybe what we call a biosimilar, we shouldn't be calling it a biosimilar, and we should call it a biomimic, because we know that in some countries, there are commercially available agents that are copies of the originator, but which have not been evaluated using the stringent, specifically defined criteria of the World Health Organization guideline for biosimilar, okay? And these agents should be called intended copies or biomimics, okay? So they don't follow the strict regulations that we would like all the other drugs that we use. They have followed that, but these ones, they don't. And let me tell you, there are several companies out there with massive pipelines. This is Boehringer Ingelheim, a company that has nothing to do with ophthalmology, but they are doing a phase 3 study to evaluate the efficacy of a bevacizumab biosimilar. Look at Sandoz from Novartis Group. They're copying etanercept, rituximab, adalimumab, and infliximab. So this means biosimilars are part of our future, okay, or near present anytime soon. Good quality biosimilar. So biosimilar is not a bad word. Biosimilar is a great thing that will lower price and make drugs accessible for everybody. Look at Samsung. They've never been in the drug world. They've been building phones and TVs and other electronics, and now they have the largest pipeline of biosimilars coming into the market, including bevacizumab and ranibizumab, okay? But what is the difference with other biosimilars that are available these days? They are undergoing massive very long phase 3 clinical trials to prove that the two Ferraris work in the same way, that the two complex molecules, they have the similar performance in this type of population. So what do we know about the use of ranibizumab? In this case, we may select other drugs and do the same thing, that they have multiple indications. For each of these indications, we will have specific trials in different subpopulations, many sub-analyses of these, largest amount of patient data. So this means zillions of patients being treated with these drugs. So we know exactly what is the performance, what is good for and what is not good for. And maybe the trials that we have for the biosimilars that are put in our hands these days are very, very limited. A 3-year follow-up doesn't answer the question, how many patients are late responders? What is the rate of atrophy if I use the drug in the long run? What is the sustained effect? Maybe at 3 months, there's no immunogenicity, but at 6, there's a lot of immunogenicity, and we say, it happily works because I've done 3 injections, but then 4, 5, and 6 drop out completely, and we are not allowed to go back because there's a lot of immune response from the patient. So maybe a small trial like this is not the best way to prove that a biosimilar is exactly the same, or a biomimic, we should best say, is exactly the same as the original drug. So some may say that, how come? This is approved by the government. It's the same situation that I get from colleagues in Argentina. They come to me and they say, well, but it's approved by the government. Why should I bother? Government should be taking care of us, right? This is a crash test. This is what it is used with cars that are being sold out there to evaluate how safe they are. And they give a star grading. If a car gets 5 stars of safety, it means you can go ahead and crash. No problem, nothing will happen to you, okay? But if a car has a 0-star rating on safety, it means that you highly likely die above 60 kilometers an hour, okay? So we think that the government is really taking care of us. This is the list of cars from Korea, Japan, France, that are being sold, in this case in the Indian market, and let me tell you, we have a lot of these in South America as well, that have 0 stars, and the government is really happy with these cars being sold out there. Why? Because they sacrifice safety for a lower cost. So they want to make sure everybody's got a car and dropping safety a little bit. That may be happening with some of the drugs that are put in our hands just to drop the cost. The thing is, we need to know exactly what the cost is. So we need to have a cost-efficacy calculation. If we have a biosimilar that costs half of the original price, but works only 50%, well, that is exactly the same. You get 50% for 50% of the price. It may work 70, and we can work out our calculations for the long term, okay, and select a population. That's why we need to push. We want data. That's what we should be saying. We want data from these trials, not 3 months of 100 patients. The future is that we're going to have a lot of drugs coming from countries that are not US or Europe, other options for treatment, new molecules like brolucizumab, lower priced availability of drugs that have been there for a good while, and let me tell you, in India, you have the lowest price worldwide for this drug. So, but for all these drugs, we may have solid data supporting the use of these. So we may conclude that we should be actually humble regarding what we don't know, and maybe regarding certain drugs, we don't know exactly what is the efficacy. Being wrong may have some effect on patient safety. Innovators, that is, original drugs, they do have detailed information regarding each of the conditions and the way you treat all the patients. And we should make sure that the pathway for the biologic approval is not a simple one that one size fits all, but it's a stringent way of getting drugs into the market. Actually, companies that achieve that, they're going to control the market because they're going to come with an equally effective drug at a lower price. And that will force a price war and really drop the price down. So that's why I'm saying biologics are a good thing, but we need to make sure they don't slip through the net. They just go through every single door that we need to test them. So just to conclude, I know that in some regions of India, everybody are big football fans. And what if you want to bring one of these players, okay, our Lionel Messi from Argentina that plays in Barcelona, and you want to bring this guy to play here in India, at one of your clubs. And they tell you in Barcelona that if you want him, you have to pay 300 million euros if you want this player. And I come to you and I tell you, no. Don't spend 300 millions. I can get you this for 300 euros. Okay, this is a guy from Turkey, looks similar to Messi. Actually, what he does is he walks in the Rambla of Barcelona and he gets pictures with tourists for €5, okay? And I tell you, it looks the same. And actually, I have tested them and they've done a little race and they're equally fast. Is that enough for you to say €300 or 300 million euros? No. You actually want to make sure this guy performs in the same way. And the only way you may know this if they play several and several tournaments and matches. Always making sure he wears the Barcelona t-shirt, because when he wears the Argentina one, he doesn't work. Okay, thank you.
Thank you, sir. I would invite Dr. Preeti Manjunath, our director and phaco-refractive surgeon, to give a memento to Dr. Patricio, and Dr. Sri Bhargav Natesh to speak a few words.
I thank Dr. Patricio for such a wonderful talk. Thank you, Doctor. You've been traveling tirelessly, I understand, and you've taken time. He's traveled from Guwahati directly. He's come from the airport to give this talk. We need give him a wonderful applause.
Thank you. Thank you.
I have a couple of questions for him. If audience have any questions, they can ask. Okay. There are few interesting things that came out of the subgroup analysis of rise and ride. In India, we face a lot of these problems where people come to us with very, very high glycemic indices. We see 10, 12, 15, and then we also have their postprandials, way too high. So generally, we have been looking at the postprandial sugars to come down before we start treating them with anti-VEGFs. But we don't necessarily look at the the glycosylated hemoglobin to come down as such. So one interesting thing that came out of rise and ride is that if the glycosylated hemoglobin is more than 8 or less than 8, it did not make such a big difference for the resolution of the macular edema. So I wanted your personal opinion on that. That is my first question. My second question is, I think we are in very exciting times, and this is the first time probably we are seeing a situation where diabetic retinopathy can be reversed. I understand that there is a cost factor, and at least in India, we are not yet ready to kind of replace laser for the treatment of proliferative diabetic retinopathy with injections alone. So is there a PRN kind of an approach which we can adopt for at least those flat NVs, the small NVs, and how do we follow up these patients?
Okay, excellent. I'm going to try to answer those questions from some analysis from the Ride and Ride study. We were very lucky. The Ride and Ride is an FDA trial that was originally planned to run only in the US. Recruitment wasn't going very well, so they came down to Argentina asking for support, and we managed to recruit 19 patients in 3-4 months, so we did very well recruiting patients. What the Ride and Ride trial did, they did something that is called stratified randomization. You put a mark of 8% of A1C, so you make sure you have a proportion of patients above and under 8 in each of the groups, so you can calculate whether the sham or the treated groups behave in the same way, whether above 8 or under 8, up to 12. The maximum was 12. Other trials is 10, this one was 12. And what we have seen is that baseline A1C, this is baseline A1C, was not a predictor for outcome in the long term. It was further done for other clinical parameters like lipids, hypertension. None of these parameters correlated with success or not, which means you don't need to wait for those parameters to normalize to start your treatment. It doesn't mean they can take the sweet, what's the name of this little thing that, well, they don't need to take these freely because it doesn't matter, 8 or 12 is the same. That's not the point. What I'm saying is, I'll see that patient with 12 A1C, I start treatment today, and I'll make sure they lower while they are being treated. In some places, they don't start treatment until the A1C gets under 8, and it means wasting a lot of time, okay? So for several clinical parameters, not only A1C, but also looking at other clinical parameters, it make no effect at the beginning, okay? This is baseline. Some patients during the patients that we managed during the trial, they end up doing, we're talking about almost illiterate people, you know, that they draw conclusions very rapidly. And she was saying that she was being cured from diabetes because her vision was 20/20. So, and I said to her, you need to make sure her A1C was 17, the highest I've ever seen. So she was not bothered with diet because her vision was perfect. And that was the only issue she had. Risk of heart attack, she couldn't be bothered. Risk of stroke, she's not bothered. Renal function, she goes to the toilet and she comes back and according to her, it all works, okay? There's fluid coming, that's it. That's all she needs to know. So some patients, they start going in, you know, that direction. So don't do any evaluation. The better the patient is treated, probably the long-term effect is going to be better. Let's remember that we're going to have to manage this patient for several years. The other point was, you said that you were not prepared to start treating patients with PDR just with injections. We are not either, okay? All we are showing is a biologic effect that never happened before, okay? So this could be a trend. In the US, where they have big insurances, good coverage, good systems of SMS, patients come back, pick them up with an Uber, and very easy to have a close follow-up of the patients, they do it for a small patient population. What we have is a change in strategy. For 40 years, the menu for treating PRP, for treating DR, had only one dish, which is PRP. Now, there's a second page of premium treatment that may work for some, okay? And in these cases, we know that the magic happens. It's difficult to apply, it's very costly, and it will maintain, after Right and Rise, we did a PRN extension phase. During that extension phase, after it, we saw that 75% of the patients held the regression rates for 2 years at least, and 25% of them started walking backward. That means developing diabetic retinopathy again. But the future will bring us drugs that in wet AMD, they work every 12 weeks. And highly likely in DME, they work every 16 weeks. That means possibly maximum treatment will be 3 injections a year, okay? 1 every 4 months. And what if we can maintain that with 2 or just 1? So it's just a change in paradigm. It's showing something is happening clearly, but it's not for everybody. These days, 99% of my patients that come with diabetic retinopathy beyond the 47, 53, they get PRP straight away. Subgroup of patients that they are at this level and have DME, I may postpone the decision a little bit just to make sure is this one of the patients that respond better? And I'll give you a bit of data from the presentation that I'm going to be presenting in your retina. The biomarker that will tell you whether a patient will have regression or not is the response to the DME. If a patient has DME at baseline and at 3 months DME goes away, it's highly likely that if you do 3 more injections, you will have massive regressions of diabetic retinopathy. If you've done 3 and DME is still there, unlikely this patient will have massive regressions up to 6 months. So you don't have to treat them all. You do 3 injections and you see, has DME gone away? Yes, that means I will do 3 more and I will induce this regression. So that was the strong biomarker for predicting which patients have regression and which don't. But it will take 5 years until we drop the lasers. In the meantime, we're doing a lot of laser. I don't know if I managed to answer your questions. Thank you.
I had one question for you. Do you use ranibizumab or bevacizumab, or because the cost difference between the two being so much, okay, that the same issue of the cost comes in? Do all the studies Rise and Ride and the reversal of the diabetic retinopathy status, what you have shown, such dramatic results, is the same thing going to happen with the use of bevacizumab? Have you seen that in your practice?
Probably, but less. You see the response of protocol T, that bevacizumab was similar but not the same. Like when you go to China and you go to these markets and you buy things and you ask, is this original? And they tell you, original but different. Okay? So I think bevacizumab is the same but different. Okay, so the CAT trial, for example, comes to show that these two drugs have non-inferiority between them, okay? But the CAT trial was done with a non-inferiority margin of 5 letters. And that was an academic trial. If you try to do, and I am part of the executive committee of a trial that is running for a new drug from Roche these days, and if you try to get the FDA to approve a non-inferiority trial with a 5-letter margin, they start laughing at you from the moment you open the door. They tell you 5-letter margin. That's, you know, you could be as fast as a Ferrari running if you say, okay, I'm going to go from here to there, but the Ferrari needs to be on the other side before I do 20 meters. You can do that walking, okay? So I think that CAT study shows that the non-inferiority margin was too wide, and that is why bevacizumab and ranibizumab ended up being non-inferior. If that would have been a FDA trial, the limit would have been 4 letters. It's usually half of the total gain. That is where you should put your cut. And no trial, only the Anchor trial, had a gain of 10 letters. All the others had 8 or less, right? So that's what Ivan used, 3.5. So if you recalculate these trials, you will see that there is similar effect, but there's no non-inferiority, okay? But if you compare the cost, this is very, very cheap. You just give it a go. And Protocol T shows something very similar. The situation in Argentina is that for some patients, we have to use bevacizumab because we have no options. And I truly believe that even if it is risky because I don't know where it was compounded, we don't have compounding pharmacies like in the US, so I don't know where the drug comes from, okay? Who did the compounding? Who was it maintained in the fridge? I don't know any of that. Sometimes no treatment is actually worse than any treatment. And I just discussed this with the patient. Some others, very few, may have good coverage of very, very expensive drug because the cost is about $1,500 for, that is $1,500 for either Eylea or Lucentis. Okay, so these drugs are very expensive. Some people have insurance coverage, and in that scenario, I don't mind using any of them. But there's no worse treatment than no treatment, and I have a lot of data regarding the performance. Poor, but I can do my I can do my own cost efficacy calculations because I know exactly the performance. Protocol T, CAT, ILLUMINE, several trials looking at bevacizumab. Some other drugs have no data whatsoever.
When you ended the first talk, you said that the dryness of the macula is not very important when you consider treatment. But say, for example, if the patient has a 6/6 vision and has macular edema, we tend to treat that patient, right?
I think that if there's fluid in the retina, that is a bad thing for the retina, okay? Be it a little bit, that is just an increased thickness, or being a lot of it, that means large chunks of cysts affecting the retina. If that retina has 20/20 vision, I could take a few months to make sure if patient can normalize, adjust a few parameters to lower those values if it is a treatment-naive patient. Beyond 3 months, I would start treatment anyway, because we've seen in the Right and Right study that the longer you delay the trial, the treatment, the worse the outcome. If you start too late, you will never get the result.
So basically,
now, what if the patient is already being treated? Sorry. What if the patient is already being treated? I'm not saying that it doesn't matter. I'm saying that you still need to fight for that fluid to go away, but the patient will be sort of happy because vision may be already picking up.
Now my question is now, when we saw the breathless analysis, they said that if somebody is not responding to anti-VEGF, you keep on injecting after a certain point of time, they would have good vision in spite of having edema.
Yeah.
Right? And when you ended your talk where you said that, if there was a combination between ranibizumab and ranibizumab alone, ranibizumab plus dexamethasone and ranibizumab alone, although the vision was same, but the one with the combination had lower
thickness of the
retina.
Yeah, dry retina.
So going back, applying the same logic that we did with 6/6 vision and some amount of edema, don't you think treatment combinations are better and keep their macula as dry as possible? Wouldn't that be an acceptable kind of outcome at the end?
Combination therapy for a patient with 20/20 vision would be possibly too aggressive.
No, I'm not saying for that. I'm saying applying the same logic that we treated a patient who had macular edema at 6/6 because there was fluid and there was change in the OCT, right?
Okay.
But when the breathless said that, you know, after a certain point of time, it doesn't start responding, to leave it in spite of having the edema. But there was the trial that you showed that, you know, where
you
yeah,
shows thinner retina.
thinner retina.
So your question is that should be an important thing.
Shouldn't it be?
It may. I don't know. Truly, I don't know how to answer the question because I don't know. Maybe that retina is like excessively dry. Or maybe the long term of having a dry retina is great and actually the combination therapy truly works. What we would need is long follow-up of these patients taking part in the protocol U and see whether these 12 months, just difference in thickness but same vision, stays for the long run and has any benefit in the long run. So I don't have a trial.
Reducing the vision if they are observed like that for some more time. The basis is that they have.
Sir, you can. It's working.
It could be. It could be. You may be right. You may be right that we need to make every effort in our hands to get rid of all the fluid. But you may be wrong as well because we don't have the extension of the trial.
Yeah, that's true.
That's what I'm saying. We don't have the evidence pointing in any direction. We have 12 months results showing combination doesn't give you any better vision but dries the retina anymore. What's the value of that? We don't know. We will have to, you know, measure it on a scale. What's the effort and costs of those extra 50 microns? If after 5 years means gaining a lot more vision and having a more stable, beautiful retina, well, it was worth the effort then. But if it's not, it's just a dried retina. But I take your point.
Yeah.
I originally said that retina should be dry because it has an impact in vision, and these treatments one gives a drier retina than the other one.
I mean, come back to the same question. Sometimes I get a feeling that, you know, we are doing OCTs on these patients every visit. Patient, you have treated them, you know, whether it's BRVO or diabetic macular edema. Patient has improvement in vision. Patient has 6/9, 6/6 part vision. You have given them 2 injections. The vision doesn't change. There is a slight edema still present. And I feel we are probably over-treating them. In earlier times, we didn't have OCT. You know, there was no difference between 125 and 175 microns. Are we sometimes over-treating our patients in order to get the macula totally dry? The vision is good. The edema is not going away with, you know, one more injection. So why not just leave them alone? The vision is good. The edema is, you know, as long as it maintains, it doesn't increase. I have a feeling that with the OCT and all that, there is an element of over-treatment that is happening in our, you know, in our practice.
I agree with you. I agree with you. But also, I think that maybe if we look at the, there was a sub-analysis putting all together several trials, Protocol I, Protocol T, Restore, and they show something that is very important. The more aggressive you are on the first year of treatment, the less injections you're going to need in the second and the less injections you're going to need in the third. And if you're good, maybe fourth and fifth, you're going to need no injections. So these trials, they all agree that first year should be somewhere between 7 to 9 injections, something that is difficult to achieve, okay? Second year is going to be somewhere between 3 and 5, and the third between 1 and 3. And maybe you will need an injection or not in the fourth year. So there is light at the end of the tunnel, and it's not a train coming, okay? It's truly something good at the end of the tunnel for these patients. If we treat them aggressively at the very beginning, they may see the light and do not need any further injections. But at the beginning, I think we're trailing behind that. That means doing 3 injections in the first year, 3 in the second year, another 3 in the third, and it's 3 forever. And we never induce these walking backwards. You know, we need to push these diabetes that is coming forward at great pace, stop it and send it backward. And that needs a lot of effort in the first year. If we maintain that rate of injections over the long term, Ride and Rise as an example, it's too much treatment. We don't need that. We don't need that. I tolerate the treatment very well, but we don't need to be that aggressive. Actually, if you look at Protocol I, Protocol T, what they say is as soon as you achieve 20/20 or near, or thickness less than 250, call your patients every 16 weeks and you will be fine. So that is roughly what I'm doing these days. That is an extreme to an extent. If I do a couple of injections and the patients achieve these, I said, okay, I'll see you again in 4 months. Take care of yourself, make sure the diabetologist is taking care of them, and I'll see them less often. And I just keep the ones that I need a lot of kick at the beginning. And some may never get it, because I think there's a point of no return, where some diabetics, they've been trailing with no treatment or macular edema for so long that they have drill, loss of photoreceptors, vascular damage beyond repair. The vascular damage is such that it's not able to reabsorb whatever fluid is out there. So you can either, put them fit up on a tank of ranibizumab mixed with Eylea, mixed with whatever you want to use, and those patients still are not going to do well because they cross the line of no return. And we've seen that in patients receiving renal-pancreatic transplant. They have a new pancreas, they have an A1C of 5.5, they can eat whatever sweets they want, and they're fine. Still, they have macular edema that comes back, and they need injections and injections and injections. So some patients may be beyond repair. But if we're good at detecting them early, treating them early, we will see the aggressive treatment, 7, 4, 2 injections, and no treatment after that.
So in our few case series, we have a macular edema treated with bevacizumab and ranibizumab. The edema is not responded. Actually, when we changed to Eylea, it responded very well. Instead of continuing the same like ranibizumab, like the changing with the other molecule, like Eylea, actually I have 10 patients we have treated with bevacizumab and ranibizumab, not even significant reduction of the macular edema. All the case who were treated with Eylea had a very good response, and also vision is also with a very good response. Sir, you told if you are continuing with ranibizumab, continue the same drug for a thing. Instead of that, going for implantable dexamethasone, can we change to Eylea? It has a good response, sir.
Okay, I trust that the data that you're seeing is actually true. You're seeing that. But highly likely is related to all you're doing is changing from A to B and never B to A, because you have no single patient that you have studied in Eylea, and if they do not respond, you switch them to ranibizumab. And this is a failure, a design flaw that is everywhere with all the switching papers. All the authors of these switching papers, they truly believe in the data. But if you look at this data under the microscope, you say, you're lacking a control group. Most of the time, and I tell this to my residents, what is clinically significant or statistically significant may be invisible to my clinical practice, okay? And one good example for that is smoking. Nobody detected that smoking would induce a lot of other diseases like cardiovascular and pulmonary diseases just because they were just sitting at their office and they said, all my smokers are dying and the other ones are not. That didn't happen, okay? It's because a study was done in the UK that is called the British Doctor Study. They were evaluated, all doctors across the UK registered with the GMC with the form sent over the post for several years, and that study went on for 40 years until somebody realized, look, there's a rate of cardiovascular and lung disease higher in the group that are smoking than are not smoking. Actually, lung cancer is a rare disease among smokers. Not all smokers die of lung cancer, but if you look at lung cancer, it's more common in patients that are smokers. So all these statistical tricks will affect my evaluation, particularly I'm just looking at 10 patients and I've switched them from A to B, but not from B to A. Okay? I'm not saying that your data is not right. Recently, I recruited patients for a study. 16 patients were recruited for the study. The first 14 continuous patients were affected in the left eye. What if I come to you and I show you that series and I say, possibly the fact that the heart is on the left, it means that there is more flow that goes directly to the eye, you will just have a drink and leave, right? You say, I'm wasting my time with this guy. So sometimes small series may fool you. And I do an exercise with my residents using a dice. And I ask them to throw the dice 18 times and make a note how many times they got a 1, 2, 3, 4, 5, and 6. And several of these, of my students, they would say, I never got a 6. And one would say, I never got a 1, or I never got a 4. Or the 6 came out 5 times. So I asked them, you should write a paper saying that the 6 is an uncommon number when you throw the dice. Because in your series of 18 throws, it never came out. You know how many times you need to throw the dice so you have non-statistically significant difference between 1, 2, 3, 4, 5, and 6? 1,000 times. So if you do your calculations, most of the time, the papers that we read are all underpowered to answer the question that they're trying to answer. They just solve it by putting a little line at the end saying, well, the study wasn't empowered to resolve this question, maybe another larger trial should be done, in very, very small print, okay? And this is what happens. And all we're left with is post-hoc analysis of large trials, which again is not the total truth, because it's a post-hoc analysis of a trial that was designed to answer another question. And that comes to a problem that we have in medicine, that is, trials, big trials, are designed to satisfy the FDA, not the clinical use of the drug. So they first approve the drug, and then they do other trials to see what is the performance. Okay?
Thank you, sir.
Pleasure.
And interestingly, all the case we gave earlier, no? Next when we gave the bevacizumab and ranibizumab, it is working that time. Actually,
you should have said that at the beginning. So it works as a boost. Okay, so you should put in, when you do your report, say this is the ILLUMINE boost. Okay, thank you. Thanks very much.
Thank you, sir. We are running short of time. We'll have quick case discussions. I request Dr. Anand to come to the stage for his first presentation.
So I'm going to present a few case series on dome-shaped maculopathy. Is a patient, female patient, aged.
So Dr. Pukraj Rishi will be, Dr. N.S. Muralidhar will be chairing this session, and Dr. Santosh Gopikrishna and Dr. Venkatesh Kadri will be the moderators.
Thank you. I'm going to present a few case series on dome-shaped maculopathy. The first case is a patient aged 32 years, presented with floater in the right eye since 4 days. Not a diabetic hypertensive, she's myopic. Her best corrected visual acuity in the right eye was 660 and the left eye was 6/9. Anterior segment intraocular pressure is within normal limit. The fundus examination shows a tongue-shaped hypopigmented lesion in the juxtafoveal region with superiorly few punctate lesion. OCT shows a dome-shaped macula with subfoveal CNV. And fundus fluorescein angiography in the early phase shows a few leakage. In the late phase shows increase in intensity of the leakage. And this is a left eye photo, shows within normal minute. And she was diagnosed as a dome-shaped maculopathy with CNVM, treated with the injection ranibizumab. After the first injection, her vision is improved to 6/9 with the reduction of the lesion. After the second injection, her vision is improved to 6/12 with a significant reduction of the CNVM. There is one more patient, female patient aged 37 years. She presented with a diminished vision in the right eye since 2 months. She had already received 1 dose of intravitreal ranibizumab given 3 weeks back, not a diabetic or hypertensive. Her best corrected visual acuity in the right eye was 6/24 and the left eye was 6/6. Anterior segment and intraocular pressure is within normal. In the right eye, fundus shows more than 1 disc diameter subretinal fluid with few subretinal deposits in the right eye. And FFA shows few punctate lesion over the margin of the subretinal fluid. And ICG not shown any polyp. Then OCT shows a dome-shaped macula at the foveal region with SRF fluid, and over the lesion there is a SRF fluid. She was treated with a multiple dose of intravitreal ranibizumab. Each time when we were giving, she was responding and recurring the fluid. We did a micropulse laser also. After micropulse laser, fluid is reducing, it is recurring. Later, she received 2 doses of intravitreal Eylea. So each time her vision is improved to 6/18 and the fluid is reducing. See, my question in the dome-shaped maculopathy, is there any role of PDT in dome-shaped maculopathy? Thank you.
I don't have any personal experience in doing PDT for a dome-shaped macula. Do you have any?
If anti-VEGF is working, so why go to another treatment?
What I presented. Actually, we tried with both eccentrics, like ranibizumab. We tried micropersuasive. Then later we tried IU also.
Okay.
After all these things also, she is recurring the fluid. Is there any chances if we do PDTs then the thing just
It's an excellent case, and it shows a condition that is very disappointing to manage. The first case has a neovascular membrane and has the funny shape. The majority of the pure dome-shaped maculopathy will just show fluid on the apex of that curve and nothing else, and there's nothing you can do to get rid of the fluid. You know, we've done several injections, no injections. We tried a lot of things, and if there's no neovascular membrane, you will have no effect whatsoever. Actually, we had a meeting with Frank Holtz once, and we presented some of these cases to him, and as soon as he saw the first slide, he said, don't you ever inject any of these patients. With the caveat saying, I have injected many, and we said, yeah, we did the same. Don't waste your time, don't waste money. Pure dome-shaped maculopathy responds to nothing. It's an anatomical glitch. It's a break in the structural shape of the sclera and leads to this pseudo-fluid accumulation and never ever responds. You can, the first one responded with increased vision and getting rid of fluid because there was a membrane on top of it. But the pure case should be the one that is just fluid, and it's just fluid because of the poor situation of the retina on top of a broken sclera, kind of, you know, these dome shape that we see.
But if dome-shaped maculopathy, if they had a complication like SRF fluid and CNVM, we should definitely treat, no, sir? Like,
if there's something else, yes. And one thing it is important. We tend to do the OCT of the patients with fluid under the fovea. We tend to do just horizontal cuts. And sometimes the dome shape is only appreciated if you do vertical cuts. So I remember the day we showed this case to Frank Holtz. We showed the horizontal cut where you could not see the dome shape. As soon as he saw the case, he said, do you have the vertical cut for it? And I knew that he had resolved the case straight away, because I showed the vertical and it was a dome shape and said, don't inject, don't do anything. So the recommendation is when you see funny fluid in a patient that is not that myopic, sometimes it could be like two diopters only of myopia, you know, and most of the reports, they go from 2 to 10, so it could be any range. You do your both vertical and horizontal cuts, so you may pick up that there is this change in shape of the dome-shaped macula.
Suppose if I have not treated the first case with any injection, what?
But no, the first case had CNVM.
Yeah, the first case had a
CNVM. What sir is telling is that you know, if you have the dome-shaped macula with just the fluid, with no demonstrable CNVM either on OCT or on FFA, then we are wasting our time trying to tease these patients.
Yeah. And it responded to a certain point, because that's the fluid that got rid of with the injection, but the rest is due to the dome-shaped macula. It's a very, very good case. Very good. Congratulations.
Dr. Sweetie.
This is again another entity which got discovered only by the OCT, like myopic foveochiasis, this focal choroidal excavation. So all new diseases are coming by using newer image modalities. You know, I don't know what more. We would have many more of these, I'm sure of that, in the days to come. As we image these structures better and better and better. So we're going to have a dilemma when, you know, we're going to have the disease before it causes any symptom, and then we'll be in a dilemma, should we treat, should we not treat at that particular stage.
True, and we're going to have more, even better imaging devices. And once we sort the total conundrum with the OCT, then will be the OCTA with, you know, extreme definition, and it will be difficult, yeah.
Good evening. So presenting a case of a 60-year-old lady who came to us with a complaint of blurring of vision in her right eye since 1 year. She was known diabetic and hypertensive since 15 years and well controlled on treatment. So in brief about her past medical history, in December 2017, she had some blurring of vision in her left eye. At that time, the visual acuity was 6/6 in the right eye and 6/9 in left eye. And she was diagnosed as having both eye PCV, and she was treated with the PDT in left eye followed by anti-VEGF injections. And later on, she was treated with anti-VEGF injection in the right eye also. So when she presented to us, her best corrected visual acuity was 6/12 in right eye and 6/6 in the left eye. The amsler shows metamorphosia and the rest of the anterior segment was normal. So the fundus of the right eye showed fundus of the right eye showed some pigmentary changes in the peripapillary area with the dull foveal reflex, the corresponding autofluorescence, and the left eye showed some pigmentary changes along with some whitish subretinal deposits in the peripapillary area, the dull foveal reflex, with a small hemorrhage near the disc. I'm not able to. Yeah. So here is one small hemorrhage. There was a small subretinal hemorrhage. So the OCT of, we did the OCT, so right eye showed SRF at the fovea, and the left eye OCT showed dry fovea with the peak-shaped PD along with the SRF in the peripapillary area where the lesions were present. So we went ahead and did the FA and ICG. So the right eye FA in the early phases show mottled hyperfluorescence with the leakage in the late phases, and the ICG of the right eye shows the corresponding corresponding area of the FA showing branch vascular network with the polyps near the fovea, which is showing washout in the late phases of ICG. And the left eye FA shows some mottled hyperfluorescence with the leakage in the late phases. And the ICG also shows the branching vascular networks and the polyps in the early phases, which shows washout in the late phases. So we diagnosed the patient as having both eyes peripapillary PCV. So my question is, shall we treat both eyes together, or shall we go ahead with the right eye first, because patient is symptomatic in the right eye and she is not complaining of any decrease in vision or blurring of vision in the left eye? And next is, shall we observe the left eye and treat just right eye? And if we are treating the patient, so shall we treat only the monotherapy of anti-VEGF or the combination therapy?
This patient already received the PDT to the left eye in December 2017, about 9 months ago. And then the left eye seems to be a flat macula, no fluid under the macula with 6/6 vision. A right eye, which had only anti-VEGF injection, seems to have recurrence now with the fluid under the macula. So in this scenario, what should be our choice? Should we go with PDT to right eye with anti-VEGF, or PDT to both eyes with anti-VEGF? What would be your
okay,
approach?
so we know that PCV is a type of neovascular AMD that has a lower drive of VEGF. So that means they wouldn't respond to anti-VEGF, but they will respond less to anti-VEGF than any patient with normal wet AMD. If you want to support your decision whether to go for monotherapy or combination therapy, you have trials that justify both the situations. You can see the DRAGON trial that does monotherapy treatment with ranibizumab, or you can look at the PLANET trial that does the same thing with aflibercept. Yep. Or you can look at the combination therapy, pure, strict combination, which was the Everest-2. The Everest-2 shows better results regarding polyregression, lower number of injections, and that is why the use of PDT, it takes more time, it's more laborious, and the patient, you need to do more things to the patient than just the monotherapy, but you will induce more polyregression, possibly a better outcome in the long term. But you may live in a situation like, I don't know, I know that in this country you have a lot of experience with PDT, but in our country, for example, is not available PDT, neither is ICG. But we have a lower rate of PCV. So we treat those patients with monotherapy. I will need more injections, probably I will need 10 every year, so that means a heavy load, and I will reduce slightly the risk of the two things that we fear the most, which is the massive serosanguineous hemorrhage that we may have in these patients. So that may be related, and it's still not very clear how much we reduce by the time of the amount of reduction of polyp regression. So I think we should look at each of the eyes of the patient independently, treat them both accordingly, and if they have active disease, you may want to do treatment to reduce the risk of, to induce some polyp regression to reduce risk of complications in the long term. And always combination therapy appears to work better, although you have to do more effort than just monotherapy.
Would you choose between aflibercept and ranibizumab? What's your...
I think things are being presented in a funny way because of the following. Every time they show us two trials in which they want us to see which drug is better than the other one, they will show us the total gains in vision. And they will usually show you the following. They show you the Planet study showing gains of about 10.5 letters, okay? And if you look at the Everest 2, the gains were 8 for the combination therapy, although very few injections, only 4, and the monotherapy were 5 letters, okay? So people say 10 is better than 5, I should use aflibercept instead of ranibizumab. Then when you adjust for baseline vision, that means the poorer the vision, the bigger the gains, you see that there is actually no differences there, okay? And an example of that is the Dragon study, which baseline vision was very poor, and is the study that gains 12 letters, okay? So until we have a head-to-head, I'm not totally satisfied, although there are several Japanese trials pointing in one direction, I'm not totally satisfied that is one drug better than the other one. I think any one that you would choose, you will do better, and the only one that has proven the superiority of the combination is the, from the Everest to the ranibizumab. The aflibercept in the Planet didn't do combination therapy, just did rescue PDT, and that was only done for very, very few patients. I think it was like 10% of the patients. So we don't truly have, at least not even 2 similar studies. There's a great review that was recently, it's a simple and very good review that was published 3 days ago by the group from Singapore and Australia, Mark Gillis and some other authors, and they did a great review of all the data available. I just forwarded it to here, so we can have everybody can have that trial. It's free online, so you can get it, and it's a great review of how to manage these patients. I think the best approach would be to take treatment.
Where was it published? Which
Yeah, I'll show it now. Let me see. Yeah, but it's a great review, and it puts all the trials. Laptop, Planet, Dragon, every single one of them, so you can appreciate what was the starting vision, what was the final vision, and how much effort you need to do. And you will make your decision, and according to the conclusions of the paper, according to how many resources you have. Because I'm sure it's not the same working in Amrita, when you have an expert doing PDT, where you're working in somewhere that you don't have access to it. So you just put in the scale, what are the options that you have, according to the resources that you have, and you make the decision how to treat that patient.
And one more question, sir. Is there any role for focal laser for the PCV, like in this peripapillary PCV?
I'm not aware. I mean, I know there are a few reports that they say that peripapillary, you can just kill them with the laser. Yeah. But not big trials, just, so I'll keep it with a traditional treatment, I should say.
Sir, any role for micropulse laser? Sorry, any role for micropulse laser?
I haven't seen any data from micropulse, but I mean, micropulse, they're using it for everything these days. It's like acupuncture, it works for.
One of my friends said, the good old days, they were doing, incising the optic nerve sheet decompression.
Yeah.
So he said, this is a surgery that's hunting around for indications.
Yeah. We have the technique now.
So I think micropulse laser is something like that. It's just hunting around for indications, for it to be it's been around for such a long time, but still we don't have.
It leaves less damage to the eye than the RON, the decompression of the optic nerve. So.
Thank you.
Thank you, Dr. Sweetie. I would call upon Dr. Arpita.
Okay. It's very simple and very good. There's a table with all the
Mark Gillis, is it? Mark Gillis is.
Good evening, everyone. I'm Dr. Arpita Pereira. I'm a fellow at the Vitrio Retinal Department in Narayan Nethralaya, and I have 2 cases that I would like to present to you. So coming to the first case, it involves a 73-year-old gentleman who has been coming to our hospital since early 2012. He was being seen at the glaucoma department where he was diagnosed with the primary open-angle glaucoma and put on treatment for the same, and then he presented later on in November 2012 to our department, basically for diabetic retinopathy screening. So his best corrected visual acuity at that point of time was 6.6 parts in the right eye and 6.6 in the left eye. His anterior segment showed some early cataractus changes. The fundus showed mild NPDR in both the eyes. However, in the right eye, we had noted some RPE alterations and some subretinal hemorrhages just temporal to the optic nerve head. We decided to do an OCT, and this is what we saw. There was a lesion noted just temporal to the ONH, a PED, along with some subretinal fluid. We also got an angiography done. Unfortunately, I don't have the images. There was an occult leak present, and so we made a diagnosis of a juxtapapillary CNVM and proceeded to treat the patient with intravitreal Avastin. 1 month post the Avastin, we noted some amount of response, but not to our satisfaction. We repeated the Avastin a couple more times, but as you can see, the PD did not resolve and there continued to be some amount of subretinal fluid. We decided to repeat the angiography again. However, at this point of time, we noticed some areas of hyperfluorescence on the FA as well as some areas of hypersenescence on the ICG, which we thought could possibly be polyps. We proceeded to treat the patient with an anti-VEGF along with photodynamic therapy. But to our dismay, 1 month post the PDT, the patient's OCT picture looked like this. There was a large collection of subretinal blood along with a hemorrhagic PD and possibly a rip that had occurred. So we decided now to proceed with an anti-VEGF injection along with pneumatic displacement of the subretinal blood. And luckily for us, the blood did clear up and his visual acuity again improved to 6.9. We advised him an additional repeat anti-VEGF injections, but the patient refused the same. However, he presented 15 months later, this time with drop of vision in the right eye, and his best corrected visual acuity now was 6.18. His OCT looked like this, with a large PD present inferior to the fovea as well as another one present temporally. This was in September 2015. So we proceeded with an anti-VEGF injection. 1 month post the anti-VEGF injection, you can see that there is some amount of response, but it is not to our satisfaction. Again, repeat injections did not make much of a difference. We thought we would try angiography once more and see if we're missing out on something. And this time, again, you can see that the polyps are well made out, and at this point of time, you can even make out a well-defined BVN, or a branch vascular network. So we figured we would try PDT once more along with anti-VEGF. However, the patient was not willing for PDT at that point of time, so we just had to make do with the anti-VEGF. And luckily for us, this time there was some amount of response. And as you can see in Jan 2017, the height of the PED had come down to a certain extent. But our success was short-lived. You see the next month again, patient presents with a large PED inferiorly, along with some subretinal fluid. I'm sorry, the video is not playing. We repeated the FA and the ICG. And you can see the polyps here have migrated even closer to the fovea, and the branch vascular network is very well delineated at this point of time. Even on the OCT, you can see the split in the RP, which correlates with the presence of the BVN. So we proceeded with an anti-VGF and a PDT this time as well. Unfortunately, we didn't see much of a response. And as you can see, the height of the PED did keep increasing. And this was in Jan 2018. So the last time around, we did a repeat ICG. And as expected, we continued to see polyps. However, this time the branching vascular network was very well delineated. So finally, we decided we would go with an anti-VGF. We advised PDT once more, but the patient, due to financial reasons, could not undergo the same, and we thought we'll try our luck with focal laser. And luckily for us, it responded quite beautifully. I'm sorry, the video is not playing, so I can't show you the other sections.
I think I should congratulate you and the patient.
Actually, this was a
real lot of ups and downs, and still the patient is continuing to come treatment with you. That's really lucky. Yes.
So the problem was actually the advancing edge of the BVN, and the polyps were just going on. So what I did is I just blasted the entire BVN as well as a polyp
with a focal laser.
Focal laser, almost like a modified grid tight packed. So just hoping for the best.
I think right from the beginning, the thing was PCV. It was a peripapillary PCV. And somehow response to Avastin for PCV has been very, very poor. In my experience, either I use ranibizumab or aflibercept. If at all I want to combine with PDT, I would rather use ranibizumab or an aflibercept with PDT rather than Avastin, because somehow the response to Avastin is very, very poor.
The thing is, financial issues, the patient was a little, I had to kind of...
yeah, so sometimes you have to do what you have to do with what you've got.
Yeah.
And you ended up doing something that more or less worked. Very lucky patient, because she could have had a massive bleeding and the whole thing be gone. But she was lost to follow up, came back, and he was always lucky to have just fluid and not a massive hemorrhage.
True.
So, very good case, very good case.
There's another case.
I could proceed to the second case. So this is a 71-year-old gentleman who presented with progressive diminution of vision in the left eye for over 10 years, and he presented to us basically because of drop of vision that he noticed in the right eye. He was diagnosed as having ARMD elsewhere and was already on antioxidants. So on examination, his best corrected visual acuity in the right eye was 6/9 and the left eye was 1/60. Nuclear sclerosis of grade 1 noted in both the eyes. The fundus photograph, it's not too clear, but on the right eye there was some RP alteration seen at the macular region with a small PED just temporal to the fovea. And in the left eye, we noticed basically scarring involving the macula. So this is the OCT of the right eye. You can see some subretinal fluid along with the PED present adjacent to it, and an ERM as well. And this is how the left eye looked, with predominantly scarred CNVM with some fluid present subretinally as well as subretinally. So we proceeded to do an FA and an ICG. This is of the right eye. There's some stippled hyperfluorescence, but no obvious leak that was noted or polyps or BVN that we could make out on the ICG. This was the left eye. Showed some areas of staining because of the scarring. We could make out some areas of hypersenescence on the ICG, which we thought possibly could be a membrane. So we proceeded to treat the left eye with anti-VGF. We used razumab here. And 1 month post the anti-VGF, you can see that the fluid had all resolved in the left eye. However, the right eye, you can see that there's an increase in the amount of SRF as well as the height of the PED. So we proceeded to treat the right eye with anti-VGF. Now this is 1 month post the anti-VEGF. You can see there hasn't been much of a dent when it came to the reduction of the SRF. And even on a repeat injection, there wasn't much of a response. Similarly, we repeated the FA and ICG hoping to find something. We could see some amount of a spectral fluorescence present in the FA, but again, no clear-cut BVN or a polyp that we could see. We thought we would try once again with an anti-VEGF. So after 2 repeated injections, if I can just run the OCT. You can see that there hasn't been much of a difference with the anti-VEGF injections. Repeat FA-ICG, which was done recently in May 2018, showed again some amount of speckled hyperfluorescence on the FA. We could see some amount of hypersenescence on the ICG, but then again, we weren't sure as to whether it could be a possible BVN or not. The patient was maintaining, this is again 3 months later, you can see that the amount of SRF has increased marginally. And however, the patient continues to maintain a vision of 6-9 paths. So our question to you is, what do we do next?
So thing is, there was no definite leak, just knuckle leak. I was wondering whether... OCT I have done, sir. I couldn't see anything. We did, I did. Yeah, nothing actually. So thing is, I was just... Nothing. Actually, we couldn't retrieve the data. There was some issue with the motherboard in the OCT. So the thing is, I was wondering whether it was an early PCV in making, or, I mean, I even switched the anti-VEGFs in between. Patient was only particular on one particular thing. So
actually, you have more questions than answers for this patient, which is you have a lot more experience than me in treating patients with PCV, and this is a clear case in which you look at the OCT and has the double layer.
It looks more like a PCV, although ICG has been done 3-4 times.
And nothing.
It hasn't shown the typical polyps, but although in the last ICG, you see the network of blood vessels, but no polyp. I wonder whether repeated injections have changed the morphology of the PCV. That's one thing we have to keep in mind, because this patient has received a lot of anti-VEGF. Yes. Yes. So I would think that this is one case where you could try aflibercept, because in patients in whom ranibizumab hasn't really shown, we have found that aflibercept does tie up these and gives good results. You could think of that, but I don't think I would do PDT in this eye, because I mean there are no polyps, or the blood vessels are not looking that active for me to try PDT here, and vision is also good, 6/9. So I would be a little worried with the one-eyed status. I would rather try aflibercept here than doing PDT with an anti-VEGF. That would be
How about pecchicoroid? Did this patient have pecchicoroid?
Didn't look like, actually. Didn't look like. I even checked the EDI.
Yeah, doing an EDI is a good thing, because under the area where you have the 2 areas of PED, you should have a thickened
but
choroid.
How about the sub-RP hyperreflectivity, what he has? Almost like a PCV as a
then if there is pecchicoroid, PDT has a lot of chances of giving him, at least morphologically making the retina normal, because, CSR, all patients respond pecchicoroid. And if you treat it like, even if you treat it as CSR, it's okay. Yeah. So I would do PDT combined treatment.
Why not treat this patient like how you used to treat occult CNVs once upon a time? Because the vision is 6/9. The older dictum was whenever you have an occult CNV, you observe till the vision is
observe and treat only if it worsens.
No, that's what I have been doing actually. I'm not
only if it worsens, because the evidence on the coronary of the other eye is that he has a membrane which is more of a CNVM rather than a PCV in this particular patient, because the ICGA is not demonstrating any polyp, first and foremost. Second one is that he has a CNVM in the other eye. And in this eye there is no polyp, then why do we even think that this is a polypoidal vasculopathy rather than an occult CNVM?
My only argument against that would be, if you have an agent that is shown to reduce the fluid, why not give that benefit to this eye?
Yes.
Yes. That's That's what I would. If you didn't have anything, okay. I mean, you had no other option.
But did the vision increase and decrease, or is it the same?
Vision has been the same.
The same.
He just doesn't complain. The problem is the fluid keeps increasing whenever he comes. I mean, I usually treat and extend. I mean, most of the time I'm just observing. I'm giving him a drug holiday kind.
Over a period of time also it's behaving like an occult CNVM.
Yeah, it is behaving like, I mean, even if you see the speckled hyperfluorescence has increased over a period of time.
That was what was probably, you know, pushing towards the occult CNVM part rather than the PCV, you know, because throughout all the 3 FAs you have demonstrated that it is showing only speckled hyperfluorescence rather than any polyp or any CNVM.
OCTA just doesn't show anything, that's it.
Thank you.
Thank you.
Thank you, Dr. Arpita. A small announcement. There is a PDT machine kept for demo at the back. Anybody who is interested or who has queries can speak to the speakers or to Dr. Sri Bhargav Natej. Cases from our retina OPD which is pertaining to today's talk. Here is a 54-year-old male who presented to us 8 months ago with diminution of vision, 8 months ago again. He had a best corrected visual acuity of 612 in the right eye and 6.6 in the left eye. He was basically referred to our hospital for a fundus fluorescein angiography. Until then he had not received any treatment. And this was on presentation in the last November 2017. Right eye macula showed a large PED in the macula with a subretinal hemorrhage and a large subretinal grayish membrane along with exudates. So a clinical diagnosis of choroidal neovascular membrane was made in the right eye. And the left eye showed very few retinal pigment epithelium changes at the fovea. And on FA, the early phase showed certain stippled hyperfluorescence, and as the fluorescein angiography proceeded, there was pooling of spirotemporal to the fovea, and there was a leak inferotemporal. So a diagnosis of CNVM was made and patient was referred back. He received 2 injections of ranibizumab there in their hospital in the month of November and December. Recovery was not satisfactory, so patient presented back. He received 3 injections of Eylea at 6-weekly interval, January, February and April. But BCVA improved to 6-9 parts in the right eye, and later he switched on to bevacizumab. But the clinical picture was fluctuating with subretinal fluid reducing post-injection and again picking back after 4 to 6 weeks after injection. And one more thing on the OCT was that dense hyperreflective echoeclasion above the PED, which did not respond to any of these injections. This was in June 2018. This was even before starting the Avastin, and this is in August 2018 after Avastin. Only the subretinal fluid has been increasing or decreasing, but the CNVM complex as such has not responded either to Eylea or to Avastin. And this is the comparison picture between January 2018 and the top one was in August 2018. So this is the picture. So I would just want to ask, where should we stop or where should we refer a patient for ICG in such cases? Because this is the common clinical scenario what we see. On FA we do see a CNVM, but when do we ask for an ICG? Because patient has responded to our treatment but has not responded completely.
I think this looks more like a PCV. I mean, PCVs do respond variably to your anti-VEGF agent. Some of them respond to Avastin, some of them don't respond, and this patient has had Avastin, Eylea, everything. He showed some response to Eylea, I think.
He definitely showed response to Eylea, but again after 6 weeks, after the post Eylea, he returned back to the same.
Yeah, I mean, even Eylea needs to be given, 4 weekly or 6 weekly. So that is, one injection is not going to really solve. The other thing is, do an ICG. If it shows a polyp with a BVN, you can consider PDT with an anti-VEGF. Probably that would give a longer remission in this patient.
It's not that anti-VEGFs do not respond, and anti-VEGFs take care of the symptoms, they take care of the fluid, they take care of, the swelling most of the times. So even in our setup, the patients that we get, they've already received a lot of anti-VEGFs before they come to us. Anti-VEGFs buy time for these patients. You tend to gain time, even if after maybe 2 years, since the start of the problem, the patient comes to you and he's had like 12, 13 injections. The patient really responds after PDT and gets very good vision because in between, for these 2 years, anti-VEGFs have taken care. They have taken care of the, the edema and the real health of the neurosensory retina. So, your anti-VEGFs have been responding, but I would suggest PDT and combined treatment after this.
Patient has been now counseled to get an ICG and to opt for a PDT or a combined therapy, PDT with the anti-VEGF injection. And this is a second case of a 72-year-old lady. She is a patient with us for the last 6 years, from 2012, complaining of blurring of vision in the right eye. The best corrected visual acuity then was 6/9 in the right eye and 6/9 in the left eye. She deferred FFA initially because she had multiple drug allergies, and based on OCT analysis only, the treatment was started. And this was on presentation in the right eye. It showed a PED along with a massive subretinal blood which was healed, and also a PED and few RPA changes. That was the OCT picture on presentation with a subretinal heme and intraretinal edema along with few exudates and a large PED. On the left eye on presentation, there was foveal thinning on the OCT along with few RPA changes and loss of the ISOS junction at the fovea, and vision has been maintained at 6/9 to 6/12 in the left eye so far. Right eye, the disease has been increasing. Since 2014 to 2018, she's been receiving anti-VEGF injections on PRN basis. A diagnosis of IPCV was made. But what made me present this case was in the left eye, in 2017, superior nasal to the disc, there were few RPE changes initially, but now it has been showing some subretinal fluid along with some exudation. So is this a peripheral exudative hemorrhagic choroidopathy? What should we offer to the patient? Should we offer PDT with combined therapy, that's PDT combined therapy in the right eye along with PDT in the left eye for this? Or should we just do a focal laser and leave it? Because this patient is a hypertensive, she's not on systemic anticoagulants, but we have had 2-3 patients who were on systemic anticoagulants who have presented with a massive subretinal hemorrhage or vitreous hemorrhage with the PCV changes in the other eye. So how do we go about in treating the left eye of this patient?
Usha, I would do an angiogram in this patient. I think that's the basic thing you need to do to study what is happening in both the eyes. Whether that will give you the answer as to whether the left eye is the peripheral form of the disease what you're looking at the right eye. Even in the right eye, if you see, it's more away from the fovea rather than near the fovea. See, this could be PEHCR, that's possible. So do an angiogram, and then whatever it shows, then you can probably take it forward from there. And the left eye where the lesion is in the nasal side, and focal laser also could be an answer. But if you are doing PDT, you can as well switch over to the other eye and give some laser, and if it doesn't work, you can always come back and do a focal laser. But do an angiogram, ICG angiogram, FFA and ICG, to study what is happening and learn what's happening. And then you can base your treatment based on what it shows.
Okay, sir.
I have another question, which is, how do you manage these days? I mean, if any patient with a suspected wet AMD in whichever form, you will do ICG to all of them, or you will start by FA treatment, FA and OCT treatment, and if it doesn't respond well, then you will revert to doing the ICG.
We don't do ICG as a routine.
Okay.
We do FA, we do OCT. FA is done most of the times as the baseline. And if there is any suspicion of PCV, you do ICG.
Okay.
If you think it's not PCV and probably straightforward AMD, you go ahead and treat with anti-VEGF therapy. If the response is not as per what you expect, then you do an ICG.
Okay, excellent.
Thank you, sir.
Thank you, Dr. Usha. Now, I would like to call upon Dr. Vidya for her presentation.
Dr. Vidya is here. Only one. The last. The last but not the least.
Dr. Sharina, please be ready for next presentation.
One more, last. Where is she?
Good evening, everyone. 45-year-old gentleman complains of decreased vision in right eye for 6 months. He was diagnosed as CSR and underwent laser treatment twice elsewhere. and he was using steroid cream for hands for some allergy, and he was not a diabetic or hypertensive. Coming to ocular examination, right eye best corrected visual acuity is 6/60 and left eye is 6/9. Anterior segment was normal. Right eye fundus showed ILM folds in the macula and subretinal fluid near the superior arcade with bullous exudative detachment inferiorly. Left eye showed multiple PDs in the posterior pole. Then we did OCT. OCT of the right eye at macula showed neurosensory detachment and subretinal fluid in near the superior arcade with neurosensory detachment inferiorly. And left eye showed PD changes. Then we did FFA and ICG for, in the early phases there was hyperfluorescent with pulling in the late phases. It's the same in ICG in the right eye. Left eye showed window defect. Then B scan of right eye, there was subretinal fluid with fibrin superiorly and inferiorly there was an exudative retinal detachment. So diagnosis, right eye multifocal CSER with exudative retinal detachment was made and left eye PD. Then we started on a trial of tab eplerenone of 25 mg per day and we asked the patient to review after 2 weeks. After 2 weeks, the visual acuity improved by 6/24. I'm sorry, I don't have the fundus picture. And there was decrease in the subretinal fluid and macula showed minimal SRF with neurosensory detachment. And that time we increased the dose of eplerenone to 50 mg and we asked the patient to review after 1 month. The visual acuity remained 6/24 with a few RPE changes superiorly and in the macula. And the inferior exudative detachment has decreased. There was decrease in the fluid. And this was the OCT in the recent visit with minimal SRF. So my question is, what next?
I mean, there are so many papers and case series reported out there that they treat CSR with PDT, with melatonin, with eplerenone, and this is a condition that sometimes comes and goes, and there could be a coincidence of two factors happening at the same time. I don't have a preferred treatment for managing these patients, and sometimes you wait and it goes away, and some others they get chronic, and fluid never ever goes away. So I still struggle to.
Can I ask Dr. Indu if you want to do PDT for her, for this patient, where would you do the ICG? Where would you do the PDT? He has a funny lesion in the superior arcade. We don't know. I mean, what that is. That is anything contributing to the whole thing?
My question to you is, has the patient stopped using the steroid ointment?
Yes.
He stopped using.
He came to us, we asked him to
yes. Most of these problems, they come with continuous use of steroid ointment. It's a steroid responder. So I would suggest just wait for some more time. Now that he stopped using steroids, so I think let's wait, and it's already coming down. Yes. So I think you should wait for some more time, unless of course you feel that your neurosensory retina is really thinning. If it is thinning, that means we are losing vision in the long run. So then I would suggest that you do PDT. In such kind of a patient, I would not do. I would not do. No, there is no need. I think we have, the cause there. So if you ask the patient to stop steroids, he should be better.
Sir, how about repeating the FA and ICG in this case? Because I have seen some multifocal CSRs, done some PDT and focal laser as well, but they recurred with new sites after 3 months, 6 months.
Even after PDT?
Yeah. Even after PDT. Not at the same site. It's a different site. There were focal leaks, especially at the fovea.
The steroid
No, these patients were not on steroids. These are multifocal CSR in both the eyes. So since there were leaks at the fovea, I had done PDT along with focal lasers to multiple leaks which were extramacular. So what had happened is the CSR recurred. It came down completely and recurred after almost 6 months or 1 year.
So you did PDT and those focal lasers at the same time?
Yeah, no, focal lasers and PDT at the same time.
At the same time?
Yeah, focal lasers I had given it only to the areas of leakage.
No, but if you're doing PDT, then at least wait for 1 month for the effect, and if need be, then perhaps that focal laser, which I really don't believe in, because with focal laser you are.
Which one, sir?
Why both?
Why
no, there were
both?
multiple leaks at multiple places, not at just subfoveal leak, extramacular leaks, just with the focal laser.
When you have multiple problems somewhere, do your absolutely, and also you can do your PDT in the center, then in the periphery again do it at the same sitting.
Yeah, but 5 is
same
maximum
sitting.
size, and if
No,
you
you
go on
can
giving it, so
you can make 2 circles, one for the center, one for the
because
sides.
that patient
and,
at least had some 7 to 8 leaks at multiple places, just not 1 or 2 leaks. They were everywhere literally. So I had to go separately with focal lasers. I had to literally see that it blanches on a focal laser.
When you do PDT, even if it is low fluence, you are creating more VEGF. And by doing argon laser at multiple, you are really creating VEGF. You are, and when so much of VEGF you are creating, then it's better than give an anti-VEGF also.
Even a focal spot, does it?
You are creating VEGF. When we do PRP anywhere, at least in.
No, CSR.
No, whenever you
CSR,
do something, you do something to the retina.
When you do the laser, you may be inducing, you're breaking the Bruch membrane, and actually that's the animal model of neovascularization. You just grab whatever animal model you have, and you do argon laser to the retina, and everything will start neovascularizing after that. So the sort of
combine when you are treating CSR, do you
I don't. No.
to extend the same logic?
No, I'm just talking about so many argon laser treatments he's giving.
Okay.
Because of that, I'm saying.
He's breaking the Bruch membrane and may induce.
So these are the spots what we usually give even in other case. Yes, but DME also.
It's a very mild.
Yeah.
It's not like we're completely. Yeah.
Ideally.
Yeah.
No, but aren't you afraid in treating milder areas when you're doing, say, for example, what case he's describing? Say, if you have extensive areas of leakage in multifocal CSR, you know that the lesions are, if you put them all together, is about, 7 or 8,000 areas. So are you not afraid of having an RP?
When you have a very large spot for PDT, the effect of laser gets decreased. But once you have a small spot, that's a very focused, and that area gets a lot of laser effect. So there is a lot of chemical reaction in that small area. So when you increase your spot, it is not that your, the laser spot or the chemical reaction increases manifold. It actually gets decreased over a large area. So doing a small laser and, having it act there or having a chemical reaction for 83 seconds in a smaller area is actually equivalent to having a large spot and exposing it for 83 seconds. It's the same thing.
The intensity of the light that the laser emits, whether you keep the spot smaller or
that is just a visualization light. The laser that you are emitting has no light. That is a pointer. It's a red light. That's a pointer. You can increase or decrease. That is just for you to visualize where you are giving your laser.
Thank you.
Thank you, Dr. Vidya. We'll go to the next case.
Dr. Sharina, please. Dr. Shilpa, be ready.
Good afternoon, everyone. I am Dr. Sharina Thomas, Vitreoretinal Fellow in Narayana Nethralaya. So here we have a case of a 59-year-old male patient who presented to us with painless progressive decrease in vision in the left eye since 4 years. He has history of treatment with anti-VEGF injections in both eyes, 5 doses over the past 2 years. On examination, his best corrected visual acuity in the right eye was 6/9 and the left eye was 2/60, not improving. Anterior segment examination showed early cataract in the right eye and he was pseudophakic in the left eye. On fundus examination, he showed a large hemorrhagic PED temporal to the fovea along with some subretinal exudation superiorly. In the left eye, he had scarring at the posterior pole with an admixture of old and fresh subretinal hemorrhage. OCT was done and the right eye showed pigment epithelial detachment in the subfoveal region and temporal to the fovea along with some subretinal fluid. In the left eye, the OCT showed mainly scarring at the posterior pole with intraretinal cystic areas and some pigment epithelial detachment temporally. FFA OCG was done for this patient, and in the right eye, he had this hyperfluorescent area temporal to the fovea corresponding to the hemorrhagic PED and some stippled hyperfluorescence at the macula. In the ICG also at the macula, it was mainly hyposenescent with no area suggestive of polyps or branch vascular networks. In the left eye, FFA OCG showed areas of scarring and multiple hyperfluorescent areas which suggested polyps and BVNs. So the question here is, in the right eye, whether to observe or treat, because the patient has a vision of 6/9, and if treat, what is the role of PDT here? And the left eye, whether to observe or inject, since the patient is 2/60 in the left eye.
Patient right eye as of now is asymptomatic, but because of the PED elsewhere taken 5 injections so far, but no change. The only concern what patient has, will my right eye become like left eye?
He has taken 5 injections to the left eye.
Left eye or right eye also. The PED has been treated so far with the biosimilar ranibizumab. So right eye is concerned about right eye now. He's asymptomatic, but show that OCT right eye.
I thought the early phase ICG of the right eye, just at the fovea, it showed some hyperfluorescence. If you go back to the first one, the first, ICG, FA ICG. Just stop here, stop here. There, what is there near the fovea there? Isn't that a bunch of polyp or a BVN? Just at the fovea, you see the hyperfluorescent,
yes,
hyperfluorescent.
Go next.
Next.
Is it?
You do have something there in the fovea. I would say, you know, it's probably if you have not, I would not treat the left eye. I think left eye has already reached the end stage. I don't think there is any point in wasting money on that. For the right eye, I would consider 2 things. One, switch over to aflibercept, or consider PDT with aflibercept, or one of the anti-VEGF agents, either Accentrix or...
OCT again, please. The right eye.
It has problem in the macular area.
Yes, macula is definitely...
There is a PED.
Yeah, and there's quite a thick choroid there as well. I mean, there's an increased thickness of the choroid, which I think we should be doing a lot more EDI, particularly in a normal retina. You would see the whole of the depth of the choroid, but as soon as you start having features that may attenuate the light that you have on top, the EDI will just bring the sweet spot of the maximum resolution of the OCT to the under part of the retina. So you may see whether there's a true sort of enlargement of or increased thickness of the choroid. This patient has no drusen whatsoever. What else can it be? No drusens, increased thickness of the choroid, you know.
Yeah. So definitely this is PCV. Maybe the BVN is not shown in that large PED area, maybe because of the hemorrhage. Once maybe the height decreases, BVN might become prominent. Only concern is what to do now, like as you suggest, yeah. The morphology must have been altered, yeah.
And one thing that is important here, there's no room for mistakes. There's no room for loss to follow up. This is a single eye now. This is the patient's quality of life and ability to sustain for himself depends on this eye. Is a 6/9 eye, but has a lot of features. So, as it was suggested here, you know, the full-blown treatment of probably combination therapy should be the ideal scenario for this patient. We know it works the best, the combination therapy. More effort, you know, 2 things to be done, but you know, it will be more sustainable in the long term. Need less injections and reduce the activity of the polyps.
Yeah.
I personally think that at this point of time, do we have a comparison of aflibercept versus combination? Because I somehow feel that if visual results of aflibercept monotherapy are almost similar to combination therapy.
But we don't have combination therapy
with aflibercept.
With aflibercept. It was just rescue in very few. And there was one big difference in the PLANET study, which is very interesting. The determination of what is a PCV, which as we have been seeing through all these cases is not that easy, was done by a reading center in Singapore for the Everest 2 study, and for the PLANET study, it was up to the conscience of the principal investigator. So if you were on your own and you say, this is a PCV, it gets into the study. If you think it's not, it doesn't get into the study. While with the Everest, so PLANET is a study of patients with a lot of PCV, while Everest is a patient that all of them have PCV. So that's why we need to read these 2 trials totally differently.
But still, my impression is that aflibercept works better than Accentrix in PCV. I mean, at least this is what our experience with patients with PCV whom we have treated with Accentrix and switched over to ILIA. Because of the cost involved, the number is not high, because number of patients who could afford to take ILIA is definitely much, much less. At least that has been my impression, but I can't really quote any study as far as that is concerned.
Yeah, for the same reason, SF now advised aflibercept for him. So one night, of course, ideally PDT and combined treatment.
I'm only worried about drop in vision.
The thing what I'm concerned is if you're doing PDT, both doing it if it contracts aggressively and if it drips nasally. I mean,
you may have one of these. Yeah. So how often you have these choroid infarcts or these complications related to PDT? Are they very often or?
No, about the ribs, I've just had 2, but then I'm very, very careful. You know, in a very huge PEDs, I go around it, but never, you know, or maybe I do just low fluence in the huge this thing. No, here, I would give it in the central area.
Yeah.
I would give it in the central area because the polyps are there. Yes, there is no need to go to the PED side because then you're scared of the rib.
Yeah. Now,
this way you'll avoid the rib.
One thing what I was wondering is, even if you plan a PDT at this circumstance, at this point of time, where do we do it? Because
in the center,
only in the
macular area,
only in the center.
Macular area.
Yeah. Otherwise,
and give anti-VEGF just after your PDT.
Okay. Otherwise,
don't wait for like 7 days to give your anti-VEGF because first 24 hours are crucial because that is the time we lose PED because there's so much of VEGF which comes after PDT. So just give anti-VEGF immediately after PDT.
Otherwise, would it be wise to inject a few ILIAs and then see the response?
Yes. Yeah. I mean, when you inject anti-VEGF soon after PDT, you say, do you take them to the injection room or a OT and then give? Do you use any torchlight? I mean, there's a concern of torchlight being shined on the eye.
You know, we really used to be scared. We really used to be scared of this photosensitizing thing. And what I've noticed is that once you do PDT, it takes about, say, 1 hour for the patient to really get sensitive to light. So you have a window of about an hour. And I have my laser in the operating room anyway. So I do my laser and give injection immediately. And also the dye remains in the system. We used to keep the patient in dark for 5 days. This is something like 15 years back. And now we've come down to only 12 hours.
48 hours is what?
No, now only 12 hours. Only 12 hours. If I do it in the morning at 9, by evening I discharge the patient. So it's only 12 hours now. We really don't have to be scared of, you can, we have a torchlight and we can give injection. That is no problem because you can't wait for that long. First 24 hours are really, really crucial. If you take care in that 24 hours, I guess the problems will not occur.
Thank you.
Dr. Shilpa?
So when you see a non-PCV AMD, you just hug the patient saying, oh, you have the normal one. You're the one that, with just a few injections.
I'm Dr. Shilpa from Prabhat Clinic. Today I have a case of 56-year-old lady who presented to us in 2014. She had history of metamorphopsia in the left eye. Her visual acuity during the presentation was 6/9 N6 in both the eyes. So she gives a past history of receiving 3 Lucentis injection in the left eye at Shankar Netralaya. In the right eye, she had some focal choroidal excavations. In the left eye, it was suspected of having an occult CNVM. So she received 3 Lucentis injection in 2013. This was the post-Lucentis injection in 2013. And she came to us in 2014 with SRF in the left eye. So we continued giving 3 Lucentis in 2014, and this was the OCT in the left eye after 3 injections. So the patient was asymptomatic for 2 years, 2015 and 2016. And then she came back again with metamorphopsia now in the right eye in Jan 2017. She had a SRF and few PEDs in the right eye. So we did a FA in January 2017 which showed some stippled hyperfluorescence on FA, and left eye there was a focal dot-like hyperfluorescence which increased in late phases, which was suspected of having a occult CNVM. So she received 3 Lucentis again in the right eye, and this was the post-Lucentis in March 2017. So in the left eye, again the metamorphopsia started in April 2017 with an SRF, where she received 3 more Lucentis. By July, she had received 3 Lucentis initially in that year, and left eye also she had received Lucentis, but the fluid started coming again in the right eye now. So we thought of doing a FFA ICG, which showed PCV. Her FA ICG in July showed a few branching vascular network and few polyps in the right eye, and her left eye also showed BVN and polyps. So we thought of switching the injection from Lucentis to ILIA, and she received 3 injections in both the eye, and her EDI also showed pachychoroid. So after receiving 3 ILIA in the right eye, the fluid had almost gone, and in the left eye, the fluid still persisted. So we thought of giving a 4th injection in both the eyes, and we followed it with PDT after a month in the left eye. So her left eye SRF resolved after a month of PDT, but it again started accumulating. The SRF started accumulating in March 2018, that is after 3 months of PDT. So we restarted ILIA injections in the left eye again. So her right eye, we did a PDT in actually 2 months after the last ILIA injection, and the SRF was completely gone in the right eye. This is in March 2018, this was in July, and this is in September. This is the recent one. So there was no recurrence of fluid whatsoever in the right eye. So in the left eye, post PDT, we started giving ILIA again. This was in the month of March. She received 1 ILIA. This is the 2nd ILIA injection in June, and August it was still increasing. We have not intervened after the injection in June, and this is the status right now. The SRF is increasing since past 2 months. So her visual acuity is 6/9 throughout. It is not dropped, and she is not symptomatic in the left eye. So my questions are, what should be our next plan of management? Should we repeat the ICG? Does PDT help in these patients, or should we continue giving ILIA?
Are you doing combined treatments after PDT? I mean, it's not monotherapy as PDT.
No, ma'am. Combined, but only after month of ILIA we have.
So 2 months gap?
2 months gap only in the right eye, but it has not recurred in the right eye. It was only monotherapy.
No, that is not. You know, ILIA doesn't stay more than 2 months.
Yeah,
so it essentially is a monotherapy then.
Yeah, so monotherapy in the right eye. Left eye was a combined actually, but the fluid has come back in the left eye.
I would do a repeat PDT, but you know, combined treatment. Combined treatment, definitely.
I would repeat ICG and repeat PDT with anti-VEGF, maybe preferably Accentrix or ILIA, and continue with ILIA, maybe another 1 or 2 times after the PDT, you know, till you get complete resolution. That's what I would do. I mean, we all, yeah.
Yeah, she's insured.
She's insured. Looks like.
Thank you for the challenging cases.
I request Dr. Preeti Manjunath.
Whoever said that PCV is a easy disease to treat than AMD, it's not. It's not an easy disease to treat. I think it can really test the patients and the doctors as well.
Absolutely.
And still, I think we need to await more trials to really define the role of PDT, monotherapy versus combined treatment, and the role of aflibercept versus Accentrix.
And
we still many questions unanswered in this whole area. We still don't have answers in this whole area.
What if we realize in the long term that those patients that receive combination therapy did great at the beginning? I'm not talking about drugs, but combination did great at the beginning, but the outcome for the long run was actually worse. I'm just speculating. So we need long-term follow-up of these patients. Look at the surprise that we got when we did Anchor Marina, great gains. Horizon maintaining, and then 7-up shows that patients drop vision because they develop a lot of atrophy. So we need to know, as you just said, long-term results of these therapies for PCVs.
I thank the chairperson, moderator, and our guest speaker today for this interactive session. This was really beautiful indeed. I call upon Dr. Preeti Manjunath for the vote of thanks.
So dear friends, seniors, teachers, hope it was an illuminating session with the experts shedding light and sharing their experience on PDT. We believe it has been useful to all the participants and will immensely benefit their patients. Trust, such focused meetings will educate, enrich, encourage, and engage us in application of treatments, finding a new solution, and sharing these will ultimately translate to better patient management. We are grateful to all the speakers who have bestowed this knowledge upon us. Our sincere thanks to Dr. Divakar, observer from Karnataka Medical Council. We acknowledge all the delegates present here and those who have joined us via web link. We recognize Novartis team and for their scientific support. Our appreciation to the staff of Netra Eye Hospital for a very neat job, and thanks to Mr. Ravi Kumar for audiovisual support and web link. Our gratitude to Bangalore Ophthalmic Society, Karnataka Ophthalmic Society, and Vitreoretinal Society of India for their encouragement. Also our thanks to Hotel Lalita Shok, represented by Mr. Adiman Ori and Mr. Santosh. So until next time, from Netra Eye Hospital, have a nice day. Thank you.
There will be a Retina Club meeting end of this month at the same place, Celara Diagnostics. So we will send the WhatsApp message. It will be on the last Friday of the month at 7 p.m. Okay, please do make yourself free and bring cases. Retina Club, last Friday of the month, Celara, South End Circle. I will reinforce with a WhatsApp message. Thank you. Thank you so much. Thanks to Netra team and Sri Bhargava for putting up this show. And nice to see so many people interested in this, rare PDT. These days PDT I thought was given up and,
Thank you so much. So you are the PDT expert of the country, I mean, I don't think PDT is such passion like what you say.
It was nice interaction.
Thank you, Siddharth. ಎಲ್ರಿಗೂ ಹೇಳಿ ಮೆಸೇಜ್ ಮಾಡೋದು ನಂಬರ್ ಮೆಸೇಜ್ ಮಾಡೋದು ಮಾಡಿ